Mesdames.
Ladies and gentlemen, we will start the meeting right away. We hope that everything is all right with the Zoom and the connection. I hope that everything is going fine. Dear colleagues, Mr. the General Director of the IRESA, first of all, I would like to express my deep gratitude for all the interest that you brought for this event. Thank you very much for having sacrificed your time, your precious time to move and to come here and to be with us during this meeting. We are really very honored by your presence, while of course hoping that this meeting would be fruitful for everyone, whether you are here in this room or following us online via Zoom through the video conference. The principal theme of this seminar are the biocontrol agents against the perishable foods after the harvest and during the storage. So trying to extend the shelf life. So it's an occasion to raise the consciousness of everyone because of the losses of the because of the perishable fruits which actually decay and perish after the harvest. And of course we are here to disseminate the latest results of the research realized with the partner laboratories in this Mediterranean project. The current orientation and the research of fight, biocontrol agent and fight, which are healthy and which are respectful of the environment, are one of our preoccupation. It is within this framework that our activities of research they do come in this framework, but they have to be validated at semi-commercial and commercial levels through in the storage companies and even at the level of sales, retailing sales. So this theme does not only involve Tunisia, because other countries are also aware of these problems and they participate to these efforts of research. One of the objectives of the works of research is to reduce the losses and the waste in post-harvest, actually or currently, estimated by 30%. Try to bring them to values, if possible, inferior to 15%. Of course, this is very over-optimistic, but we cannot actually reach the 0%. This is impossible. So we'll try, hopefully, to reach 15% of loss. If you have a look at the program, as you can see, you have various presentations. Normally, we are supposed to have a presentation of the Professor Gianfranco Romanazzi, who is supposed to present to us a general aspect and general idea of the project Stop Med Waste. But unfortunately, he missed the airplane and he would join us today, I think around midday, 1 p.m. So he would probably be here by then. Let's hope it would be the case. We will have also other presentations, like a presentation by Mr. Luis Palo from Valence, Spain, from the Institute IVEA, who would present and enrich us with his long experience in this field. So he will share with us his experience. We will equally have the contribution of Mr. Antonio Ippolito, who would be represented by Dr. Anna Maria Macusi on, of course, the same theme, the fighting the post-harvest decay of fresh fruit. Of course, the program would continue. We might probably have a little change and shift in the schedule. If Mr. Hisham Ben Salem agrees with us, he would give us a welcoming address. So, Mr. Hisham, the floor is yours.
Merci.
Thank you very much, Mr. Bashir. So first of all, I welcome everyone. I welcome our friends and colleagues from different member countries or member states of this project Stop Med Waste program. So here, whether they are following us remotely or being here present physically with us, and of course, we wish you an excellent stay here in Tunisia. And also for the colleagues, we hope that they would be in the future with us, among us in Tunisia. In the same occasion, I would like to thank the team and congratulate the team for me, the excellent choice of this theme that interests not only Tunisia, but also equally other countries, Mediterranean, namely countries. Only for the research, we should not limit ourselves to the system of production, but also we need to reason in agro system or food system in general. If I take the case of Tunisia, for example, for some fruits, for example, only for the bread, for example, we are at 20% or 30% of waste. So we have 20–30% of food waste in Tunisia. So we have records, I think, in Tunisia related to that. And now I've just heard the figure of 30%, which is really huge. 1/3 of the production is wasted. So this is really very dangerous and alarming. We have to say it even at the level of our system and our research. We have to work further on the post-harvest products, the post-harvest for different cultures and productions, the cereal production, for example. We focus more on the production and productivity, but I'm afraid we are not really talking about the shell life, the storage, and things that are related to other matters like the biocontrol agents, for example. That's why we should not limit ourselves in our research. Research has to be systemic. That's the first thing. And the second thing, we should not limit ourselves to the system of production. We have to go beyond the production. And there should be a certain transition in our way of thinking, in our approaches, our scientists. So we need to think about food in general as a system. Of course, I very quickly looked at the different axes of this prima, which are original axes, very interesting axes. And of course, if I really understood that this is the beginning of the project, is that it? Is that? No, we have been working on it for years, sir. So I hope that that's my bad. So I hope that we will reach positive results, but the most important thing is to disseminate and share these results, and of course to come out with everything that is valorizable at the level of the profession, at the level of the farmers and the industrials, and so on and so forth. This, to my sense, is the most important thing. If you allow me very quickly just to give you an idea about the system of IRESA in Tunisia. So I talk on behalf of my colleagues. So IRESA is the Institution of Research and Higher Education, agricultural higher education. So this is it's a system that covers and ensures the coordination between 21 institutions, 10 national institutes and research centers. So we have 10 entities, and higher education, agriculture higher education includes 11 entities, and our role consists in ensuring the scientific coordination at the level of the research. So this is what the IRESA does, and of course we ensure the administrative and financial coordination. So IRESA is a derogation of a part of actually prerogatives granted to us by the minister. So apart from that, IRESA, we are working on we work on the 6 different programs in partnership with the Ministry of Agriculture. So we are working on the chain of knowledge, particularly within these 6 projects. Of course, within that field, there is the research, there is the vocational training, there is the academic training, there is the transfer, innovation, and the validation. And in this program, we have 35 entities. We have the 21 entities of the system, IRESA. Apart from other institutes and institutions, we have technical centers, and we have the Agency of Vulgarization and Training, agricultural training. Something else, the third responsibility of IRESA is that we are coordinating ensuring coordination between the Ministry of Higher Education and the Ministry of Agriculture. So we are trying to ensure the coordination between these 2 ministries of higher education and scientific research on the one hand, and the Ministry of Agriculture. When I say a coordination between these 2, of course, here I'm talking about the training and the scientific content. This is really the content, which is a scientific, purely scientific content. This is the content of the training. Of course, our term as IRESA is to make sure that our institutes and our institutions and our researchers conduct impactful research. We don't do research or conduct research just for the sake of making research or to publish research. The end purpose is not the publication itself. We target rather the valorization of the results. Of course, Tunisia is a small country between inverted commas, and we have some expertise. We rank globally, we are 12th in the world at the level of the scientific production, I mean, which is really very important compared to the population. Unfortunately, compared to other countries, we are ranking really low at the level of innovation and valorization, and that's the major challenge that we need to overcome. So we are in a technical ministerial sectorial, so the Ministry of Agriculture. So for ministry publication is not an end per se, but the Ministry of Agriculture would like the researcher or research in general does something impactful so that it brings about the necessary change and the change at the level of the field. And that's the major challenge. So with the performance, the scientific performance, the Ministry of Higher Education and Scientific Research is more or less satisfied. But in terms of scientific production, in terms of impactful articles on ranking of the university, but for the Ministry of Agriculture, I think that we should focus more on that and would do extra efforts related to that. This is a message because, as I have said it, in the system we have 200 projects of cooperation which are in progress without actually counting the projects of financial funding projects. So the objectives is that these projects should bring about a change on the ground. So it's interesting to publish. This is very positive. We don't actually ask them not to publish, but publications should not be considered as objectives per se, but rather we should target the impact on the field, and that's the most important aspect for us. So for with that, I would conclude my speech, and I wish you fruitful workshop. And with all our support as IRESA, once more, I reiterate my welcomes as IRESA. So I reiterate my countries, the state members of this project and people who are present here. Thank you very much for your attention. Thank you very much, Mr. Hisham, for this presentation and for the ideas you shared with us and for the axis of interest that IRESA holds as far as the research sector is concerned. Now we will go to our presentations, and we will start with my own presentation. So we will start with the presentation of my work. Do you see my screen? Is everything all right? So I will start. The title of this presentation is The Progress of Stop Med Waste Project at INRAT, and the works that were conducted at the level of INRAT. For the other partners from other countries, of course, each would present the part which interests them or with which they are involved. As you might know it, the losses or the waste of post-harvest fruit. So we have post-harvest decay of fresh fruit, which is really very important. I think that I said earlier they reach 30%, and the objective is to reduce that waste or this loss, reduce it to the maximum possible, of course.
Vous êtes en train de partager votre écran. It is change our this. Tout d'abord, je veux saluer les collègues.
I would like to bid a farewell to salute the people and who are following us online. I'm sorry that the presentation could not be shared online with you. I would like to salute you, but just bear with us, so we could not share it online. So the title of the presentation is The Progress of Stop Med Waste Project at INRAT.
نقص فيه الإكرام ليش بدك شوي صباحك مانيش شوفوا لي هم تو هم شوفوا لي donc
So important quantities are are lost because of the post-harvest decay of fresh fruit. So I here have a select of pictures of decay or perishment at the level of the oranges, strawberries, and of course, they are linked to fungal species. Of course, these are fungal species which produce different types of decay. Namely, here we have the alternata. I would like to show you, I will show you some pictures of these fungal species. We have the Botrytis, which is another species, the Penicillium vegetatum and Penicillium italicum. These are the 4 fungal species over which we conducted our works, whether of identification or fight or combat. As you can see here, he's trying to go to the following slide.
Vous voyez ici l'isolement à l'état pur.
Here you see the pure state of these fungal species. Here is the alternata, Penicillium digitatum, Penicillium italicum, and the Botrytis cinerea. These are the 4 fungal species which have been isolated at their pure state and over which we have realized and conducted all the research work in collaboration. No, actually on these fruits. In this room, you have some samples of these species, of course, for those who are not familiar with these fungs and fungal species. Just to show you the importance of injuries or types of decay on the fruits and the damages that are brought about because of these species during the harvest and the harvest, and the storage, sorry. Here we have taken apples, healthy apples. We have superficially disinfected them, and then we cut them. Here you have some fruit that have been injured. This is a cut of 10 millimeters. We cut them with also some sharp devices. And here you have a healthy fruit, which means fruit that have not been cut. And there is no inoculation here. Instead of putting the fungus, we put it at the PDA milieu without any fungus, just to see the difference, the fungus actually, how they react with. Of course, we have the same cut that is here and here. And in this here, we have inoculated the fruit. And here we use the Botrytis cinerea. This is the species. The larger the cut, so we had also had some cuts with some needles, and we created like holes. And we have, of course, apples with no cuts. And after exposure to the fungals and 7-8 days of incubation, as you can see on the picture, you see the difference between the fruits which are not inoculated, but which are harmed or decayed. Cut. And so we have cat inoculated, non-inoculated. So as you can see here, there is no development of decay at the level of this fruit. Regardless of the size of the cat. This is ideal, but in storage conditions, it's not the case. Only in very, there is probably a slight development of decay or of perishment, which is really not very perceptible because we don't completely isolate the fruits in a sterile room. We leave them in a laboratory environment. And so in this case, the larger the cat is, the more the decay is more perceptible, and of course the waste is more important, and the damages are, of course, are more perceptible. So here is this to show you that with this test that we equally see with other fruits and other types of species of fungals, that the cats are at the origin or at the heart of damages, and if we need to pay attention to the fact that if individuals want to store the fruit for a long period, try to make sure that the fruit are conserved in a let's say a way that is intact, which means with no cats, because cats are the origins actually of perishment and decay. So we need to make sure that we avoid all types of cats and holes at the level of the stored fruit. I showed you the 4 fungal species. What makes us certain that these fungs are responsible for the perishment, we need to show the pathogen capacity of these agents or of these species. So we have the 4 fungal species, and we have inoculated different fruits with these fungs. Here you have the pears, apples, and even pomegranate, and also the plums. We have created inoculations with, we have made inoculations with the botrytis, the species, the fung species, the botrytis, and here we have made an inoculation probably with the Penicillium inoculum. Yes, absolutely, the Penicillium inoculum, and here with the Antellaria, I think. So we cut and we make inoculation. So our objective is to obtain unhealthy fruit so that we create the propitious conditions so that the fungus can find the necessary conditions to develop and to produce perishment and decay. And so you see that after 12 days, approximately in this particular case, 12 days later, we have a decay that is produced when we conduct these inoculations. That is a proof and evidence for us that the fungus that we have isolated are pathogens and they produce decay, and they can, of course, bring about damages. They can damage the fruits during the storage time. The same thing here by using the citrus fruits. Here, for example, the mandarin and the Thompson, the oranges and the citrus fruits. Here damages, but with no fungus, so there is no perishment. And with the different other species, fungal species, apart from the Penicillium digitatum that you can see here, and that has really brought about heavy damages on the origins, whether the tangerine or the orange, as you can see, the damages are very perceptible and visible, and they are severe. Of course, if the shell life is longer, of course, it will further decay, and we will end up losing and wasting fruit. So what we measured here, the diameter of these rods, whether for the tangerine or a type of orange. We have conducted a test on tomatoes, for example, by using these species, the fungal species. It is to show you a little bit the degree of the pathogen degree with these species, under the same conditions, and of course, which bring about more quickly a rot. Here you see the vegetatum is really very severe. The citrus species, Italicum, is equally pathogen, but it does not have the same velocity and speed of rot. of the fruit or decay of fruit. But of course, the longer the storage time is, of course, the more damaging the effect would be. Here with the Cinera, the Botanium cinera, and here the Penicillium of the tomatoes. We have here the Penicillium digitatum and the Penicillium italicum. I wanted to show this picture because it is completely the contrary to this one. You see the Penicillium digitatum is very severe. The impact is more severe on the citrus fruits, and like with the tomatoes, we have less severe damage. Yet with the Penicillium italicum, which is a very severity that is relatively feeble, have brought about further damages and rot on the tomatoes. We have noticed that particularly, and that was quite surprising to us. Once we have seen all the rot or the damages by these species, fungal species, you are probably asking yourself, how can we overcome all these problems? How can we find ways of fight, especially bio control agents, to fight this rot? So we will start with essential oils. The essential oils are extracted from aromatic or medicinal plants. They are biodegradable. They are normally safe and are considered as environmentally respectful, so environment friendly agents. I tested myself 38 essential oils. As you can see here, a table including all the essential oils. We have the scientific here names, the part from which we extracted the oil, the chemical components, and the origin of this plant or the oils from which we extracted the oils from, of course, the different corners of the world. Here I go very quickly. These are the 38 essential oils. And here, as you can see, first of all, we have conducted the test on in vitro. This vitro, so I'm trying to explain for non-specialists, when we have a milieu which we call PGA, it is a very favoring environment for the fungi to evolve, and we add in this milieu, in this environment, essential oils to create an emulsion, so which makes it more soluble inside the environment. In every in vitro, you have the impact of the essential oils. Here you have the 38 essential oils, and here you have the classical fungal species. Here it was efficient. As you can see, there is no development of rot, and here we have an increasing graduation from the most powerful essential oil to the least efficient essential oil. There is, of course, a variation amongst all the essential oils. Of course, this is the Alternata fungal species in total obscurity and favoring conditions. And so this Alternata fungal species in vitro, there is a possibility to find essential oils which are efficient to combat these fungus, particularly these fungus. It shows the inhibition here, the inhibition of Imicillium. Here, the best essential oil, as you can see, that would inhibit the fungus. The same thing here. So in this case, the essential oils, there are some essential oils who have no impact on the fungus, but others are more efficient. We conducted the same tests with the Cinetaria, and we have the same, the Cinera, sorry, we have the same variations, the same thing with the Penicillium italicum. Here to show you, to give you an idea about the fact that there are some essential oils which inhibit the increase and the growth of the fungus, and other fungus which have actually further developed, yet while in the presence of this oil. Of course, we have collected all these data to have this graph, and here we have this color. This color shows that the essential oils are efficient, and here they are less efficient. So we are at this level we have the essential oils in vitro always, which are relatively efficient against the all types of species, regardless of the species, fungal species. Now we have the cinnamon oil that was the most efficient against the increase of the growth of different species of fungus. We have the floral tools of cloves, and we have the fragrant winding leaves. These are the best, actually, the most efficient types of oils, and they have showed 89% of inhibition of the fungus. These oils would be used to treat the fruits by conducting. So this is really we need to create solutions and have these fruits immersed in these solutions. Of course, the efficiency that was observed in vitro, we need also to know whether this in vitro efficiency is the same in another environment. Of course, using these oils. These are the prepared solutions. These are the cinnamon oil. This is the clove oil, and this is another oil that shows absolutely no efficiency. Of course, if there is an inefficiency in vitro, we would have the same inefficiency in another environment. We put this fruit for 1 minute in this solutions, and we conduct an incubation of 1890 degrees, and we see the development. So we need to immerse them for 1 minute. I would like to put emphasis on the fact that these fruits, they have been cut and inoculated. We are not talking about fruits that come from the market and that we will freeze for a particular period. This part, of course, would be presented, that part of the presentation would be presented by my colleague, Ahmed Namara, and that we call it the semi-commercial period. So we do a phyte treatment of control, and we maintain in cold environments to see the development and to see the quality of the fruit and to control the quality of the fruit and so on and so forth. So here we have
we did here the fruit coating with solutions containing the essential oil, and then we added the chitosan. The chitosan was described in the bibliography as being used. It has a simulation of the defense of the plant, and it might have a positive effect. So we put it here with 1%. So it is a plastifier, and it makes the solution adhering much more to the covering of the fruit. So we added this, and then we have a control, and then it doesn't contain with distilled water. And then what we observed and about the efficiency found in vitro was not actually observed when we are in the real conditions. But for example, when you see the apples with Botrytineria, so we did it in preventive and curative manner. So I explain a bit this concerning prevention. We make the inoculation, and after 24 hours, we make the treatment with solution. So this is for curative to cure, and preventively we make the treatment with the solution, and then we make the inoculation that is after 24 hours. So the efficiency of this product should be maintained minimum within the 24 hours for the fruit. However, we noticed that this control contains no type of control relating to the conservation or preservation of the fruit. So nothing is convincing here. Same way work is done with this couple of tangerines or others. So the differences, according to me, are not really very interesting. And as you can see here, it is the same result and then the same outcome. We also spent we wanted to see if there is an effect of these essential oils concerning gas volatile phase, gaseous phase, and then we didn't find, at least in our working conditions, concerning the control, concerning witness, and concerning with the gas effect. And you see, we have put within these boxes the oil, which normally should evaporate and create an environment containing this oil. But what we saw there was actually no convincing state. So now I pass on to something else, what we call concerning the gras salts. So these are considered as being safe. Donc they are recognized as being safe. This is the present tendency to see, and we use this salt to see whether they present a positive interest for the fruit. We used as well for grass or salts here, and ammonium bicarbonate, sodium bicarbonate, and potassium phosphate. And you have here the invitations of the mission, and you see the product is efficient, and then we are close to 100%. So the metasulfide is the best with the 4 species used. The inhibition is total. This is in vitro with a concentration of 2% by 20 degrees. Now the results are going to vary according to the fungal species, and this was not efficient at all. So we continued the work in such a way to see whether these fruit are here in vivo, as you can see, and we used because in vitro we used only 0.2%, and our feeling is about this concentration. Actually, we haven't here obtained what we anticipated, so we try to increase pressure at the concentration and see whether we might have more efficiency. So we started with 0.2% with a golden inoculated with the Vaticineria. And then after 2 hours of the inoculation, we waited 2 hours, and then we passed to the treatment. So 0.05%, 1%, and then we finished with the commercial fungicide. And then we went further with 1 to 3%. So at this level, As you can see with the different concentrations, we try to work preventively and creatively. So preventively, we see that the efficiency was rather reduced and the concentration was quite weak. So what is creative? You see, we're close to 100% with 1%. So as you can see, we continued because 1% provides a good efficiency of nearly 100%. We try to limit this concentration and reach less efficiency. So for us, it's not reaching the maximum of concentration in order to get the same efficiency. So from 1% to 0.2%. So we obtained the best concentration with an efficiency, maximal efficiency. So we continued working that way. We used apples, the couple apple and botrytis area, and oranges in a certain digit. These are the most important and then the fastest manifestation of these two fruit. So in order to see whether this product sodium has any efficiency on the fruit. So what we did, we took the concentration 0.5% and then we tried to increase this concentration to reach 4%. And you see here, you have different damages, injuries, I would say, and then they are not treated, but treated with increasing concentration. So here we add 0 and then 0.5 and then 1, 2, 3, 4, 5 without any fungi. And then we put in the different fungi, and then with the important development of the fungi, and then from 0.5%, the development of the fungi was not observed. So we noticed that in this area where you have a softening of the area in the fruits, and here you'll find this in the fruit, in some fruit that are not inoculated. So this is due to the addition of the sodium, and same thing was observed with the oranges. So we might conclude that the strong concentration with the sodium metasulfide might produce a certain infection on the fruits and creating a certain softening of the fruit due to these damages. So this is the part of this area linked to the sodium metabolism, and here you have the part of the fungus, etc. Now the fungus established, though efficient with a strong concentration, can be at the same time very toxic and create this softening inside the fruit, and this will be very quickly penetrated by the fungus. I'm going to pass on to the different of the components, the compound mixture. And we used the Maltese oranges with the digitatum. So we mixed the chitosan by itself and then essential oil of cloves themselves, cinnamon oil by itself, and then we mixed the chitosan with the cinnamon oil and then with the clove oil. And then either the chitosan or So as you see, the ketazan with the sodium or sodium bisulfide, and here it is the control, the negative control, and here there is no inoculation, only to that we produce the symptoms of decay. So we see nearly what we said a moment ago. Ketazan by itself, the sodium beta sulfide and the sodium metasulfite, when used on the oranges, it is nearly equal to what we observe on the polysim. So you see the result at this level, but with the other mixtures, we don't get much. So we have the development of this problem of decay. Same thing with the apples. So we did the same thing. So you see the sodium metabisulfite, and you see the fruit are less attacked, and rather than the fruit treated with that. To conclude very quickly, so now the damages on the fruits are the first factors leading to the progression of decay. They are at the origin of an enormous loss of fruit during the harvest. In terms of the rapidity of colonization,
better tasting area is also so for a storage, all the fungal agents are capable of bringing about damages, even if they are not very quick. If the storage is long, of course, they will end up destroying and damaging this fruit. Another conclusion is that the sodium metabisulfite with a low dose is actually not has other damages. It has to be really very well adapted. Some oils, they have an undeniable efficiency in vitro. Others require a better conditions to have the same observed efficiency that is observed in vitro. And thank you very much.
J'appelle, puisque Monsieur Romanazzi.
Mr. Romanazzi normally is supposed to join us later, so I call Mr. Luis Palo, who would present to us his work and would enrich our knowledge of novel antifungal edible coatings combined with modified atmosphere packaging to reduce pomegranate post-harvest losses. Je vous remercie, monsieur. Je vous remercie pour la présentation. Bonjour à toutes les personnes présentes.
Donc, je comme de Valence en Espagne et je suis un chercheur dans le monde de l'agriculture et dans le gouvernement régional, dans le gouvernement. Comme vous pouvez imaginer, en Valence, on est de grands producteurs d'agrumes, mais nous avons aussi une grande production à travers les récoltes en Méditerranée de la Grenade et concernant ce projet Medwaste. Et dans ce travail, on a impliqué aussi un étudiant Victoria Martinez est un chercheur, un doctorant, et ma collègue aussi. Et on travaille tous ensemble concernant les produits post-récolte. Et elle est aussi une chimiste et une spécialiste dans le développement de ça. Elle est aussi pathologiste. Et vous avez toutes ces personnes-là qui nous ont déjà expliqué ce qu'il faut faire concernant packaging et l'emballage. Donc, vous savez, ça a été un travail qui a été fait en groupe et concernant les fruits comestibles, combinés avec le émail, ce qu'on appelle l'emballage modifié pour le émail.
Okay, just.
Donc, juste montrer notre localisation. Donc, la région de Valence, il y a 3 provinces, hein, Catalogne, Barcelone et Alicante dans la province de Valence. Donc, vous avez notre institut de recherche et d'interaction, juste pour vous dire que le producteur numéro 1 concernant la production des grenades, donc on a travaillé surtout, on cultive surtout et c'est dans la zone de Elche dans la province d'Alicante. Donc c'est une zone agricole. Donc, tous les jours, nous avons de plus en plus d'informations concernant le côté bénéfique des grenades. Donc, ils sont excellents pour la santé. Donc, ils ont une activité hautement antioxydante. Donc, en général, la grenade est commercialisée comme étant de fruits comestibles. Et la destination principale, c'est vers les pays nord-européens. Nous avons aussi un développement au sein de notre marché national. Donc, on a beaucoup de bénéfices. C'est une culture, donc la récolte se fait en automne vers novembre. Donc, on a des destinations d'origine. Voilà, vous avez notre logo. Comme vous voyez, ils sont pas vraiment, ces fruits ne sont pas vraiment très mûrs, hein, mais ils ont assez de temps pour mûrir. Pourquoi? Parce qu'en général, c'est moins bon. Donc, il y a d'autres qui vont vers Israël. Et ça contient moins d'acide. Donc, cette grenade est très intéressante pour le côté bénéfique de la santé avec ses composantes. Donc, nous avons un marché d'exportation très important en Europe et ils apprécient cette qualité de grenade.
Sorry. Sorry. Sorry.
the highest weight loss reduction. So the fruit less get weight loss in percentage. We identified each fruit one by one, and we weigh the fruit before and after the same fruit, before and after the storage period. So the coating is formulated with wheat wax or carnauba wax and sodium benzoate, gave us the best, the most effective reduction of weight loss. So these were the two coatings that we selected from all the coatings that we tried. You see, caused by botrytis from latent infections when the fungus arrives to the fruit in the field, and then after harvest develops what we call gray mold or crown decay in the case of pomegranates. And also we were looking at wound decays. Wound decays, as Bashir has explained it, is the fungus goes into the fruit through wounds or bruises that are produced in any part of the surface of the skin. So in this case, these were the best treatment. We didn't see any differences in case of wound decays between treatments, but for crown decay, we saw that the treatments with lowest amount of crown decay were these two treatments here, the HPMC plus the two different lipids and sodium benzoate. So sodium benzoate for pomegranates was a lot better than potassium bicarbonate. So these were the two coatings that we select for experiment 2. So in experiment 2, remember, experiment 2, we already put the fruit long-term cold storage. 15 weeks is a long time of storage for a pomegranate. So we did these two storage times, 8 and 15 weeks. And you see the weight loss during cold storage and during and after cold storage and safe life. Remember that the safe life is the simulation of the time that the fruit can take to go to the markets and arrive to the consumer. So usually is simulated for 1 week, something like that. So as you can see here, the best treatment that gave the highest weight loss reduction was the MAP, all the pomegranates stored with the plastics, with the modified atmosphere packaging in all the different evaluation periods. We looked at the gas concentration we didn't see any differences between treatments, not for CO2 and not for O2, oxygen and CO2, because as you can imagine, what is doing the modified atmosphere packaging, even if it is passive. What it's doing is modifying the levels of these two gases, CO2 and O2, in the atmosphere surrounding the fruit within the plastic. Physiological disorders, very important in the case of long-term stored pomegranates, not only Mollar de Elche, but many other cultivars. And the most important ones, rin sinking, you see, is a kind of caused by weight loss, by dehydration, is a kind of sinking of the rin, as you can see here. Pitting browning usually is caused by chilling injury. If you put the pomegranates in cold storage at a temperature too low or for too long, you can get chilling injury, which in the case of pomegranate is ring pitting or browning also. Pitting of teguments, this would be internal pitting, but in the inside of the peel. And also browning of arils, which also usually is also caused by severe chilling injury. So in this case, we saw that the best treatments to reduce the ring sinking was also the treatments with MAP, better than the ones that were only coated at the two storage periods. Also, in the case of external pitting and browning, the best treatments were also treatments with MAP. And also in the case of browning of arils. In post-harvest diseases, as before, we look for external and internal decay. In the case of internal decay, we didn't got any differences, and the amounts were relatively low or very low after 8 weeks, but higher after 15 weeks, but with no difference. But in the case of external decay, as you can see here, we got some incidents of botrytis. This is botrytis cinerea in pomegranate. This is, as I said before, it's crown decay. Usually, the disease starts here and keeps going radially from the crown to the rest of the fruit. And if the conditions are okay, I mean high temperatures and very high humidity, botrytis starts sporulating, causing these typical spores of botrytis in the fruit. Also, we found in external decay penicilliums. This picture is not from this experiment. It's just how is penicillium in the case of pomegranates. Usually Penicillium expansum, but there are other species of penicillium also, as Ana Maria knows very well, causing decay, post-harvest decay of pomegranates. And we saw that these treatments, the coating with beeswax and sodium benzoate, and in this case also the uncoated MAP, were the coatings, the treatments with less amount of external decay. What about the quality of the fruit? We look at the external color of the fruit. So we found that the external, the parameter A, well, probably I don't know if you are familiar, but the color of the fruit is measured with parameters A, U, B, and C. These are the typical parameters that have this. Pomegranates, usually the color should be around this area here. It can be more red or more yellow. So, and you see this is done by the hue angle. So A and hue angle are very important for the case of pomegranates. So we saw that during storage, but this always happens, hue decreased. So there is a loss of red color, less of red color. But at the end of the storage period and shelf life, only the fruit treated with the plastics, with the modified atmospheric, showed a little bit of higher luminosity. Luminosity is the L, is another parameter for the color. At the end of the storage period and shelf life, no significant difference between treatments were found for juice quality attributes, sodium solid contents, titratable acidity, pH, and maturity index. As you probably know, maturity index is the factor between the content, the sugars, the soluble solid contents, and the acidity. And also we look at the ethanol and acetaldehyde concentrations, and both increase during storage, but this increase, I am not showing data on this because I do not have enough time to show all the data, but we saw in the numbers numerically this increase, but when the people were trying the fruit, this didn't translate into a negative effect on sensory quality. Because as you may know, these gases are the fermentation gases. When the fruit starts becoming senescent, let's say it gets into degradation, it starts releasing these gases. And this happens when the cold storage period is too long for a specific fruit. But then this release of these gases translates into bad flavors and off flavors of when you eat the fruit. So in this case, it was an increase of the gases, ethanol and acetaldehyde, but these volatiles, but didn't go into a negative effect on the sensory quality. So just to conclude, HPMC, hydroxypropyl methylcellulose coatings formulated with sodium benzoate, this grass salt in this case, reduced the incidence of latent infections, mainly caused by botrytis cinerea, and also wound infections, mainly caused by penicillium during incubation at 20 Celsius. modified atmosphere packaging was confirmed as an efficient mean to preserve fruit freshness. In fact, was the best. It was better than the coatings, but it was better to combine them, preventing shriveling and ring-browning and reducing fungal decay, thus extending the storage life of Bollard de Elche pomegranates. The combination of the coating MAP plus modified atmosphere packaging of this specific coating, HPMC, beeswax and sodium benzoate, was the most promising treatment as it reduced weight loss and decay without negatively affecting the fruit physicochemical and sensory quality. Remember that we use up to 15 weeks of cold storage, which is a long period for a pomegranate. So this work we got several publications. So it was published in a scientific journal, and also it was used by this Italian master student to present his master at the University of Bologna. And more people to thank that collaborate in this work: Veronica, Maricruz, Amparo, and an Italian professor, Elena Baraldi, and also, of course, the funding, our project, Stop Med Waste, which in the case of Spain, because Prima is funded by each specific country that is involved in the project, is by our government, the Ministry of Innovation, and specifically by our national agency for research funding, and for this plan of recuperation of which was the next generation EU funding, which this project is involved in. And with that, I would like to thank all of you for your attention. Thanks a lot. Thank you very much.
Thank you very much, Dr. Professor.
Je remercie, professeur. Professeur Luis Palo. Ceux qui sont en train de nous suivre en ligne, je pense qu'ils sont en train là de nous voir et nous suivre. Now, what time is it now? I think it's time, high time we called. What do you prefer? What do you suggest? Shall I call Anne-Marie to present her work, or we take a coffee break and then we come back with any with that? So the coffee break. So we will go for a coffee break and we will be back.
Votre attention. Maintenant.
Can we have your attention, please? So we will go now to the presentation of Dr. Anna Maria Nacuzzi. Anna Maria, the floor is yours.
My presentation concerned innovative strategies for the control of post-harvest pomegranate diseases. First of all, the pomegranate trade is very important in the world, with a production of about 80 million of tons and the surface about 800,000 hectares. The 70% of production was concentrated between India, China, and Iran. Instead, in Europe, as previously said Luis, Spain is the most important producer, followed by Italy, that import the 4% of the global market and is an important producer. The most important cultivars are Acco, Wonderful, and Bollard de Elche. But comparing the data of total production and harvested production, we can observe a gap of about 5,000 quintals. This is due to post-harvest pathogens mainly. So we can observe that in the field, the 18% of losses are caused by abiotic diseases, such as bad handling, weather elements. Instead, the rest of the losses are concentrated between the harvest and the retail. So we add the 23% from the harvest till the transportation, and the 21% due to the transportation from to the marketing. These are the most important because these are caused by post-harvest fungal diseases. Very important are the cold storage condition because we have to attend the temperature of about 7 to 8 Celsius degrees with an humidity rate of 95%. And as previously said by Luis, the modified atmosphere obtained by microperforated plastic bags is very important, such as an arrangement in one layer within the box. In this mode, we can store the pomegranates from 4 to 6 months. However, we had the gap production in April, May, June, and July mainly, caused by the differences between the states of the south and the states of the northern that are an opposite production season. Concerning post-harvest diseases, we had the latent pathogens and the wound pathogens. The latent pathogens attack the fruit in the field during the blooming stage, and mainly are Alternaria botrytis, Coleotricum, and Coiniella genera that represented the 65% of the epiphytic population. Instead, the wound pathogens use opening of the tissue, such as wounds or lesions, to attack the fruit. And these attack the fruit along the entire production chains. The main genera involved in wound infections are Penicillium and Aspergillus that represent the remaining 35%. Important is the biology of the pomegranate because the flowers are male or hermaphroditic. The male flowers are useful just to attract the pollinator. Instead, the hermaphroditic fruit became fruit during the maturity. And these are our focus. Another important characteristic is the capellar axis, that is this one, because it's the first via for the pathogen diffusion along this line. And the division among different cavities, that is a second diffusion. So we have the pathogens that enter from the calyx, go along the capellar axis and spread in the other sides. The persistent calyx, in fact, is one of the most important characteristics of this fruit, such as the persistent stamen, that are secondary infection source. So the leather rind, the woody rind, and the amount of polyphenol compounds of the rind is not enough to control the pathogen's infection. Another important aspect to consider are agronomic practices, because steady irrigation or a nitrogen fertilization or a calcium fertilization are really important to prevent cracking. Cracking that cause secondary infections, mainly due to Penicillium and Aspergillus. Instead, the lighting is important to obtain a good color of pomegranates. Among the most important latent infections, we had alternariosis that represented the 24% of the total amount of diseases. This is due to Alternaria alternata and Alternaria arborescens that are both mycotoxigenic species. It can cause both black spots and black heart. Black spots is a disease characterized by circular black blotches in the pomegranate rind, with black, red blackish spots in the center and around a green-yellow halo. Internally, the fruit is healthy and edible. Instead, in this case, the fruit externally seems very healthy, but internally is decayed, and is the black heart. In some cases, we can observe darker rind or lighter weight because this is a dry rot, and an asymmetrical and irregular shape of the fruit. Internally, we can observe the brown and soft areas that became covered by a dry and grayish-black sporification at maturity. Second for importance is the gray mold that represented 21% of incidence. It's caused by different species of botrytis, such as Botrytis cinerea sensu stricto, Botrytis pseudocinerea, and Botrytis cinerea group S. During the development of the disease, we can observe that at the early stage, we have small and tan spots in the crown area that became softer and browner, also the areas, until reaching the collapse of the rind. Instead, at maturity stage, we can observe a gray mycelium with black spherical sclerotia and the nesting among fruit. Also, this one is a cause of secondary infection. Third is the soft rot caused by Cognella granati in synonymy with Pilidiella granati. That was the 15% of incidence. The symptoms are very similar to botrytis because initially we had the small circular spots of the rind, mainly in the calyx area. Then we had wider, softer, and darker brown lesions that at the maturity belong whitish due to the white mycelium and covered by this spherical and dark brown pycnidia. Important of this disease is that it can cause also the color rot of the plant. Less wide than other diseases is the anthracnose that represented the 5%. It is caused by two different complexes within Coletorticum genus, Coletorticum acutatum and Coletorticum glosporioides complex. Early we can observe the circular, concentric, and brown lesion with darker spots, mainly in the center. And then these lesions became sunken and soft and merge each other. Instead, at maturity, the mycelium growth is whitish and covered by orange in the case of Coletorticum acutatum, or brown acervula in the case of Coletorticum glosporioides. This is the first of our wounds diseases, and is caused by Brooklyn mold. It represented the 26%. And the main species involved are Penicillium and Thalaromyces. Penicillium that represented the 19% and Thalaromyces just the 10%. This is a wound infection that starts mainly from wounds and lesion of the rind, causing brownish and circular necrotic lesions that became deeper, darker, and sometimes irregular in shapes. Internally, we can observe brown and soft areas, and at maturity, we can observe this green or blue sporification that cover the lesion. Also, the stamens, as we previously said, could be a secondary infection source. This slide is just to show you the different species of Penicillium. In Italy, we don't have the Penicillium expansum pomegranate that named Luis previously. We have the Thalaromyces, that is this one, that morphologically is very similar to the Penicillium, and different species of Penicillium. Concerning aspergillosis, we can observe that is the 9% of the incidence. It is mainly due to Aspergillus section nigri, exactly Aspergillus tubigensis that represents the 56%, and Aspergillus welwitschia that is the 36%. Aspergillus welwitschia that is a producer of fumonices and mycotoxins. In this case, the symptoms are very characteristic and also similar to the apple symptoms because we had this concentric discoloration of the rind from yellow to brownish red colors that became at maturity stage brownish or black with this soft decay internally, so the areas are no good. And sometimes we can observe also a powdery volatile sporification black in color. According to the Italian legislation, pomegranate is a minor crop, and until the last year, we had just sulfur that was allowed. Instead, in 2023, we had also the fludioxonil, the Bacillus amyloliquefacens, the boscalid, Trichoderma species, thymol, geraniol, and eugenol. Bacillus, Trichoderma, and essential oils are allowed also in organic agriculture. It's important the time of the treatments for pomegranate. So we need to have almost 2 treatments during the blooming to save the fruit from the latent infections. And the major risk is that we can add the fungicide resistance. To prevent the fungicide resistance, it's important to interchange active ingredients and modes of action of the products. For example, we can alternate boscalid and fludioxonil. But chemical fungicides are very dangerous for the human health, the animal health, and the environment, according to a One Health approach. So the One Health approach that Stop Med Waste project favor, One Health approach induce a reduction of the pesticide and the food waste use. For the alternative control, we tested Bacillus amyloliquefacens. That is a bacterium that you can apply in the field and have different mode of action. So it acts through the competition for the space and for the nutrient, for antibiosis, because it produces lipopolypeptides such as etrins, fungicin, and surfactins that are inducer of the systemic resistance. Instead, concerning the resistant induction, enzymes that are involved in the plant defense mechanism, such as superoxide dismutase, catalase, peroxidase, can improve the production of antifungal compounds such as quinones or radicalic form of the oxygens or lignin. We tested the the Bacillus amyloliquefacens, and we compared it with the chitosan, with the Eurobasidium pullulans, and with the seaweed extracts. In this case, we can observe the difference concerning the epiphytic population of the pomegranate rind. Particularly, all the alternative treatment, except for the seaweed extract, are useful to reduce the population of the pomegranate rind. Instead, analyzing the storage, both during the cold storage of 3 months, then in the shelf life, we can observe that Bacillus amyloliquefacens can reduce by 45% the incidence of the decay at the end of the cold storage and by 35% of the incidence at the end of shelf life. We added also another alternative control that was the dipping in neutral electrolyzed water amended with different salts, such as potassium sorbate, calcium propionate, sodium metabisulfite, and sodium bicarbonate. We compared this thesis also with the ozonized water. This because this is the topic of our work package of the project. And we can observe in this case, after the dipping in these solutions, that the calcium propionate is able to reduce by 47% the incidence of the decay at the end of the cold storage, and about the 54% at the end of the shelf life. So calcium propionate is generally used as a food preservative, but is also a potential basic substance, as we tested in this other project, Eufresco. So we can conclude that good agronomic practices, so like also the steady irrigation, the balanced fertilization, especially of nitrogen and calcium, can reduce the incidence of the cracks of the fruit. and reduce the wound pathogens. Similarly, the reduction of the soil debris and the mummies are useful to reduce the inoculum that is in the field, such as the careful handling that we can obtain, for example, clear cutting the peduncle and avoiding further wounds, preserving the fruit integrity, and controlling the wound pathogens. In the field, treatments are important, but we have to remember that the repeated treatment will be during the blooming stage and are very useful to control the latent pathogens. Among the conventional fungicides, the most effective is fludeoxonil. Instead, among the alternative ones, the better is Bacillus ameloliquefacens. Finally, we can also say that optimal storage conditions are very important to prolong the storage of the fruits. So relative humidity and temperature that are cultivar specific. For example, in Wonderful, we had 6.5 Celsius degrees and 95% of relative humidity, or a one-layer arrangement in the boxes of micro wounds prevention and the nesting control, such as the microperforated plastic bags that are useful to prevent the fruit dehydration and the fruit weight losses, reducing also fungal infection due to fungi. So thank you, and I want to thank also Professor Alagui to host this event.
Thank you, Anna Maria, for this good presentation. Merci beaucoup pour cette bonne présentation. Donc, on vous offre ce trophée. Okay, thank you very much. See. Maintenant, nous passons à la présentation de Madame Mouna Benamara, s'il vous plaît.
So welcome everybody, good morning. Today I will present for you the progress of the PRIMA project, which is the efficacy of biological compounds to preserve fruit freshness during cold storage and shelf life. The plan of this PowerPoint will be with an introduction, the objective, materials and methods, the experiments done with grass salt, sodium metabisulfite, and the evaluation of the other biological control. As we know, losses during storage may reach 30 to 40%. And this is due to fungal infection, which need a control during cold storage. This fungal infection may provoke the deterioration of fruit quality and then the perishability of the fruit during storage. The main parts of this presentation will be the evaluation of sodium metabisulfite grass salt during cold storage and shelf life, and then the evaluation of biological compounds, pythium, oligodendron, chitosan on fresh fruit after storage and shelf life. Here we start with the first part, the grass salt sodium metabisulfite treatment. So the methodology, we started with the fruit which are free of wound or injury or natural injury. So here in this part, we did not inoculate the fruit or wounded them. The fruit used were oranges and apple, and then stored at 6 degrees C for 20 days, 40 days, 60 days, and then a shelf life period at ambient temperature. The sodium metabisulfite grass salt dipping the fruit was at concentration of 0.5%. In the case of treated fruit, with a control without treatment, and the immersion time of the fruit in the solution was for 1 minute. Thank you. The parameter evaluating on the fruit were decay incidence in percentage, weight loss, the physicochemical parameters, including pH, total soluble solids, and fruit hardness. Now we move to results of the first part. The storage of oranges variety Maltese. Just to remember, here there is no inoculation of the fruit. The fruit are healthy, selected, and then stored under refrigerated condition. So we evaluate the incidence of decay during the whole storage period of the oranges variety Maltese. And here, as we can see from the first diagram, that the decay incidence is increasing with the increase of the storage period. So here are the storage period at 20 days at 6 degrees C. Then when we restore the fruit at ambient temperature plus following 6 days. Here in the first period of the storage, the decay incidence either in the control or sodium metabisulfite fruit were very low. After that, reaching 40 days of storage, as we can hear, starting from 40 days storage at 6 degrees C, oranges showed an increase of decay incidence compared to the control. And here, there is no difference between the control and the treatment. But for longer storage, and mainly after the shelf life period with long storage at 6 degrees C, the decay incidence become more it's become more higher than was registered at initial dates. And this decay incidence was, as we can see, correlated with the weight loss. And here we registered slowly weight loss for both control and the treated fruit, and reaching the 40 days. And after that, following also the shelf life period at ambient temperature, we reach a maximum, weight loss. Here, for the evaluation of physicochemical parameters, including the pH and the TSS, we did not register a significant difference between the fresh fruit and then the stored fruit, either treated or the control one. And here, the pH and the TSS during cold storage are good parameters that indicate the microbial condition of the fruit that are healthy. Here, the evaluation of the hardness of orange fruit. As we can see that the fruit, when they are fresh, they have a hard peel, which is not soft, and increasing the storage period at 6 degrees C, we reach slowly soft peel texture of the fruit, and differences between the control and sodium metabisulfite become clear. At the end of the storage, and due that fruit continue respiration rate during storage, so the fruit peel hardness was best kept with sodium metabisulfite at the end of the 60 days storage. Now we move to apple fruit. And here also the fruit were not wounded nor inoculated, just conserved as healthy fruit in cold storage. We can see also the evaluation of the weight loss during storage between control and sodium metabisulfite was very low, and until the end of the storage period, and did not reach 3% of weight loss. About decay incidence for apple fruit, we did not register a percentage of fruit rotted, so the absence of decay during the whole storage period. In the contrary, what was seen for orange fruit. Sorry, again. Here, the evaluation of the pH of apple juice. Also, we did not register significant difference between the fresh and the stored fruit, and then when fruit were maintained at ambient temperature. For the total soluble solids also, we can see here that fresh fruit maintained the initial value with slight changes during the storage period and also during shelf life. For the hardness of the apple fruit, a slight increase of the fruit hardness was registered after storage and also after shelf life, and variation are seen or registered within the control and the treated apple fruit. Here, the physiological parameters or the physiological aspects of the fruit at the end of the storage period, and mainly after shelf life in ambient temperature, because maybe some of us know that apple fruit are a climacteric fruit. In post-harvest, the climacteric fruit continue respiration after being harvested from the tree. And so if a fruit continue to respire during the storage, so the texture of the fruit will be more soft. In the part 2, we evaluate the commercial biological compound that have an ingredient an active ingredient, which is pythium oligodendron, chitosan, and sodium metabisulfite for fresh fruit during cold storage and shelf life. Here the methodology, as we did for the first part, fruit were not wounded nor inoculated with any pathogens. Fruit are healthy, selected, disinfected, and then rinsed in the dipping prepared solution with polyversum, that is the biological compound that have the ingredient, the biological ingredient activity of pythium oligodendron. Chitosan was used at 0.5%, and sodium metabisulfite, after our studies and experiments in the lab, we select 0.5% to treat fruit. And then this fruit, oranges, nectarines, and tomato were stored at 6 degrees C for 30 days and 2 days shelf life at 20 degrees C, and the same measured parameters were evaluated. Here the weight loss is after 30 days of storage plus 2 days shelf life at 20 degrees C. And the weight loss is the initial weight after 30 days, and then the fruit were reweighted after 2 days. So we can see that polyversum, sodium metabisulfite, and the classical are equivalent to a classical fungicide. The fruit hardness of orange did not vary between the evaluated compounds. Here also for pH and total soluble solid, we did not register variation between the treatment compared to the commercial fungicide. Here for the nectarines, the decay, the percentage of decay presented as the number of infected fruit compared to the total fruit. Here the experiment was done at small scale with 3 fruit replication per treatment. And we registered that chitosan and polyversum had higher decay number of fruit decayed compared to Celeste Extra. And here we show that sodium metabisulfite was effective in controlling natural decay on peaches. In the photo, as here the peach, the 3 replicates of peaches treated with sodium metabisulfite. Here with chitosan, the fruit, the softness, the decay was soft without presenting the spore powdering on the fruit. As well for the hardness, didn't show the significant difference between the control and the treated fruit within this ingredient. Here for pH and total soluble solids, we can see that sodium metabisulfite has low pH on the fruit, and it preserve or maintain a total soluble solids higher even the control, which is not treated fruit. Oops. Concerning tomato treated with this compound, we noted that untreated tomato fruit had higher decay percentage compared to chitosan polyversome, and the efficacy of sodium metabisulfite was 100%. I mean, for fruit treated, tomato fruit treated with sodium metabisulfite are healthy and did not present any symptoms of decay. Here to conclude for this presentation that the apple fruit could be stored for up 60 days without quality changes and absence of decay, because apple have a capacity to preserve its physicochemical parameters during long storage. In the contrary, orange fruit had a limit of storage period to 20 days due to fungal infection with Penicillium digitatum. And the sodium metabisulfite use could be of interest under a commercial scale with appropriate packaging condition, or to be included with other ingredient to make appropriate commercial formulation. Thank you for your attention.
Merci Mona pour cette présentation. Mona, alors le trophée Mona, donc on doit lui donner. Merci pour la présentation. Je vous informe que professeur Gianfranco Romanazzi et Marwa in the airport, ils sont à l'aéroport et peut-être dans peu de temps, in a small time, they will come here.
Please, Anna Maria, come here. So, no, just to make photo with you.
Come with us.
To make
Come with us to take the photo.
To make the photo. Here, here.
Okay.
Thank you.
Maintenant, nous allons à la présentation online. Le monsieur qui fait qui arrange ça. On doit voir les collègues qui, avec Pervin de la Turquie, espérant que les choses marchent bien. Hope that will works with Pervin.
Thank you, Bashir. Hi, everybody.
شوف تراش بدها لنا Pervin.
Shall I share my screen?
Please, Pervin, now you are direct for your speech.
Okay,
You
thank
can
you so
start.
much. Thank you so much. Hi, everybody. I will make a small talk. I was very busy with my report with this project. Because of this, I couldn't prepare a long one, a short presentation I will do. I will talk about the UVC. Do you hear my voice? Is there a problem?
It's okay.
We can't hear you well. Okay. Okay, thank you. In this project, we work with the strawberry fruits, and we use for the management UVC light applications, but mainly we use this application also other fruits. As we know, the strawberry fruits after the post-harvest stage, there is lots of crop losses because of the mainly gray mold, botrytis cinerea, rhizopus, and the anthracnose diseases cause a great crop losses in this perishable fruit.
Perveen.
Hi.
Hello, Perveen.
Hello, Bashir.
Please, you can start your presentation because before it is not possible to hear you.
Okay,
You can start.
Okay,
please.
I will do it again. So it's okay.
Okay. Please, you can start.
I make it present form. It's okay?
Okay, please.
Okay. In this project, we work with the strawberries and we use the ultraviolet C applications for the post-harvest pathogen of the strawberry fruits. It's very perishable fruits and because of the latent infection of the botrytis cinerea, rhizopus, and anthracnose, it will be a great crop losses because of this pathogen. UVC light is the wavelength 100 to 2018 nanometers, and it has a germicidal and hormetic effects. It used in many studies before this, especially Wilson's and his team worked with this UVC light too much. There is lots of studies, and they found also effective on the reduce of the storage loss in many studies. We use UVC light also in our life, in our post-harvest steps, many steps. For the air sterilization, surface sterilization, instrument, equipment disinfection, and water disinfection. It's already used in this way. We use 254 nanometer wavelength for the surface sterilization. This wavelength is not penetrate to the fruit inside. Just it penetrates 50 and 300 nanometer into the plant tissue. Because of this, it's the just surface sterilization, sterilized surface. I mean. We did this study for 2 years, 1 year from Aydın, Nazilli district. We got the strawberry fruits from the organic procedures, and second year we got Denizli city. And what we did, we exposed the UVC light at different times, and we also measured the UVC doses in the cabinet, and after we applied to the UVC light different periods. We put them into the this this packed, and we put them 0 centigrade degree and 19 humidity. For 10 days, we stored them in this cold storage conditions. After this, we looked also their quality quality parameters and decay development on these fruits. And we didn't inoculate the fruit, just we let them in the natural infection conditions. First year, we found the 3 minutes, it means about 1 kilojoule meter UV light it was very effective to control the decay development. As we see, it's already very successful treatments. Second year, we did these experiments again, and from Denizli, we got the fruits, and the applications made 10 centimeters under the 10 centimeter of the under light of the UVC light. Second year results also almost the same as we did before. The low doses are very effective. When the dose are going high, the effect also decrease. Also 7 and 10 minutes didn't control the decay development. These are the pictures, control and different doses of the UVC light applications. These are the 1 kilojoule meter, sorry, and this one is very effective. These are the pathogen we obtained from the decayed fruits, botrytis, rhizopus, and also a small amount Anthracnose choletytricum was isolated. The effect of post-harvest UV light also, we look at the quality parameters, but we didn't any differences between the treated and non-treated fruits on the quality parameters. We didn't find any differences. As we know, the mechanism of the action of the mechanism of the UV light, stimulation of the defense mechanism. In this study, we couldn't do this part of the study. In this year, maybe we can do it again. It's the hormetic effect. In many studies, this also studied, and they found stimulation on the defense mechanisms. It's also surface sterilization on the surface. If there is an pathogen inoculation on the surface, it can kill the pathogen population, and for the post-harvest stage, it will be clean fruit. You can also lethal germicidal effects, also very important mechanism, actions of mechanisms. On the fruit quality, we didn't see any differences, but in many studies, they suggest to it improve also fruit quality and delaying fruit ripening. So it's UVC light maturation is delayed, and other quality parameters, they said it's the get higher. Negro et al. Works also in this fruit and these applications. They also found 1.50 and 1.0 kilojoule doses are very effective, and they found also PAL activity was very high on these doses. They also determined the defense mechanisms stimulation. In several studies, it's also suggests the stimulation of defense mechanism in plants against this pathogen, against the this application. And some studies also suggest the cell degradation of the fruit and vegetable. So it reduces cell degradation, fruit and vegetables. So it's also block the pathogen penetration. It means PAL activity, phenylammonium lias, and some phytolexin, scoporon, scapulettin, and peroxidase enzyme activities increase in this application, and in many studies it's also already studied. There is an study also, they work with the Aspergillus niger, and they found also in different doses kill the pathogen directly. The spores kill death because of this application. This is the control ones. As a result, UVC light is a good surface sterilant. It eliminates pathogen on the fruit surface. In order to all surface of the fruit to be sterilized, a well-rotating system is required. In our study, in our study, it's not rotating once. Sometimes we have to rotate by manually, but in the practical uses and the commercial, sorry, in the commercial uses, it needs a well-rotating system because of this system. So it has a significant effect on the latent infection because of the surface sterilizants. UVC lamp should be behind of the protective barriers, also to protect users. As we know, it's also harmful for the eyes, especially. Because of this, it should be there is then protective barriers between the users and the UV lamps. I would like to give this chance to Bashir and of course our coordinator, Gianfranco. Thank you so much for the listening. Thank you, Bashir. Bashir, I didn't hear your voice.
I am happy to see with us Professor Gianfranco Romanazzi and Dr. Marwa Moumni. Welcome to Tunisia, and welcome to be with us. Although you have this problem of plane, etc. yesterday. However, you are with us now. Thank you very much for all this effort. Maybe if you want, we start with your presentation on the overview. Donc, il va nous donner un aperçu général sur le projet. C'est sa première, the first presentation. Please, Professor. Donc,
Thank you, Bashir, for the kind invitation. And it is a big pleasure to be with all of you here. And hello to all the people that are following us at home. So thanks for organizing this very interesting Congress. We will try to shortly give an overview of the project. And then Marwa Momni will give some details on what as a research unit we are doing. Tunis is Stop Waste, okay. Congratulations for the nice station that you have here, and it is a pleasure to be again in Tunisia, and for me it's the second time, and anytime it is an opportunity to meet friends and to talk together and to discuss about several options. Then I will just give you briefly something about what we are doing with this project, Stop Med Waste, because wow, I can go down. Okay, very good. Okay, yeah. I'm favored by all the other talks because they told almost everything. So I will just add a few information because it is time to get lunch. So we also have to be fast, and we want to reach the coffee break as soon as possible. That I'm sure will be very, very good. And even during coffee break, we have to try to reduce the waste. So we have to take in mind that if we take a lot of things, we have to eat all of them, and all of them will be good, because if we buy a box of fruit, we have to avoid to take them into the refrigerator and then some molded, so not useful. This is part of the waste. And if you think, generally speaking, more than half of what we buy of fruit and vegetable is lost. Is lost because it is overripening, because there is decay, because we forget in the last box of refrigerator and is there. So we can give food to the same amount of people of the one that we have if we reduce of 50% this kind of waste. It will not possible to reduce the 50%, but everybody of us can do something. From the producer, because there is the kind of iceberg of food loss and food waste. So we cannot think that it is a problem of somebody else. It is a problem that involves us, because if we waste 50%, we have to produce the double to give food to the people. We do not think to this. Or we know that there is still the hunger. There is one of the lines of United Nations that it's zero hunger. We can tell it will be impossible to reach zero hunger, but we can do more all together. So we have to try to reduce these waste. There are these and we have to do it in a sustainable way, because we know that fruit and vegetable has decay, has disease, and so on. But we cannot spray every day or every 2 days, because we need to be sustainable. We need to protect the environment. We need to use safer compounds. We need to use less pesticide. And whatever we lose, we can split in 2 categories. There is something that we lose in the field, that is from the field to the retailer where we go shopping. It is called food loss. And where the consumer is involved, and we are technicians, so we are involved in both. We are involved both in the field and after harvest. what it happens from the supermarket to home, it is considered waste. And this is just one pathogen, but there is an area, and they are taking on the basket of fruit of my home. So we have to reduce this waste. We have to decrease. In EU, they adopted this United Nations mission, and they told us we have to reduce to half the food waste. We have to use less synthetic pesticide. It means that today we use the double and we have this waste. How we can do tomorrow? We have to set new strategies. And as well, we have to increase organic farming, because the consumer, I don't know if more educated, less educated, the general consumer think that organic agriculture is safer. So for the environment, so we have to increase the organic agriculture. Then, as well, the supermarket are not happy with the threshold of the law. So there is a law that ends that you can reach this minimum, this maximum amount of pesticide residues. Supermarket, at least European supermarket, no, that is not enough. I want half, I want 1/3. I don't want all the active ingredients that you can use as grower. I want just 5 or 4 or 3. This make crazy the life of a grower that need to produce high quality and at the same time without residues of synthetic pesticides. So we claim together with a list of friends. Some of them are here, some of them are online. Hello to all of you. And thanks together to all the team, we are trying, all the team that is around the Mediterranean area, we are trying to provide, we will not solve all the problem of the world, but we can just add a few drops just to improve the situation. And there is a budget of the project that is shared among the partners to reduce the waste. So at least to try to decrease of half this kind of waste that we have by reducing, as all the speakers has already told, of a certain amount of 20% the use of synthetic pesticide from the grower to the retailer, so along all the chain. So we have a list of, are called technically work packages, so activities that are run by the different units. You already heard from the different units, so I will not give into details. For example, you learn from Anna Maria and from the team of Professor Ripolte the use of electrolyzer water, the use of ozone, their combination. You learn from Professor Luis Palou about the use of antifungal coatings, and Professor Luis Palou is organizing, I guess he told, a meeting in IVIA of this project as this one in September. So if any of you wants to join Valencia, I think that you are more than welcome. It is on September, which day? 14. So save the date, 14 September, IVIA, Valencia. But it is not. Yeah. So the University of Torino worked with the biocontrol agents that can contribute. As you heard, the Cyprus University of Technology worked on the control of foodborne microorganisms. That is important, another important issue as well. INRAT produce a lot of data. INRAT is doing a very, very good job with this project, and thanks to Bashir, because you are leading a team, a very, very important team, and you are contributing very, very well to this project. So I'm very, very happy of the work that you are doing. And we are also, as a Marche Polytechnic University, doing something, trying to use the new tools, new technology, to use of sensors, and we will give you some data today, and maybe not all of them, because we have to save something for the next meetings. And at the end, if we think that we have a fruit or a vegetables, so something that we produce, we have to make this fruit stronger, because with the stronger fruit, this fruit does not ripen fast, does not decay, and so on. So we have to apply whatever it is possible to avoid the disease without using synthetic pesticide. And this is called induced resistance, with all the tools that we saw within this project and that all the speakers talked about. So this is the new tendency, because if you think to the synthetic pesticide, you have to use earlier. And you can use more, while with induced resistance, you can use when there is no disease, so you do not increase the probability to have resistant isolates. So all the colleagues talked about the tremendous amount of papers that all the teams published. They are available, so whoever wants can search, and just Googling stop and waste can find a lot of items, and we will have a lot of meetings. I can leave the PDF of the presentation, so here any of you is interested, or we can upload on the website. Can search so I will not go into detail, but all the units contributed very, very well to this topic. And as you heard also in the morning, as you will hear in the next meetings, we had a very important meeting in Limassol in Cyprus, where all the units were there and took their data, as today here. Today we are updating what we took in Limassol, and we had a specific dedicated session there where all units exposed their research results toward a list of people from all over the world. So it was a good dissemination. Nikos Sortsakis organized this meeting. Say hello, Nikos, if you are looking at us, with Panayota and with all the other colleagues. But what is important, he organized also a field trip. And when we hear a presentation, it is something that is in the middle between active and passive. In the field trip, you interact with the other people, you take questions, you talk with the other, you know colleagues, you clarify the issues. So field trips and what we are doing in the afternoon is very, very important. And what we did also in that occasion is to talk with the companies, because often happens that research and companies goes in different ways. Each one has his own idea and his own purpose, and there is not so much strong interaction. This is a very important opportunity, this meeting, those meetings, and this project, because we are here listening the need of the company to present something that may be useful for the company. Now the company has on the market something that was developed 15, 20, 30 years ago. Just to provide you an example, that is 1 out of 100. When I had the age of Anna Maria, I was playing with yeasts, and it was one yeast that looks very, very good. This yeast is the Machinicolia fructicola. This Machinicolia fructicola is now on the market as Noli. So research takes some time, but at the end takes some fruit. Of course, not everything that passed through research became and go on the market, but whatever is on the market come from the research. So this is a good opportunity for interaction of the research that tells what it is able to do, and of the company that tells which are their need. And here there are several companies involved, and I will be very happy that they interact with the researcher to see which are their need, how we can approach them, what we can do for them, and of course we can tell what they can do for us, or we can find a middle point. So this project is a way to find a middle point between the research, because in the project there are the companies and there are the research. So it is within the project, this kind of approach, and we try to do our best to take it on. After this meeting, we had a meeting with all the partners, because we mark the time, and once in a while we compare each other to see what we are doing, which are the progress, and so on. We had a very important meeting, for example, organized by Professor Ripollio on pomegranate. Pomegranate is a new tool, Anna Maria already told, and we organized this within the biggest company of Southern Italy. That this is a machine that costs, I don't know how much, but I know, but it is very, very expensive to produce juice of pomegranate with innovative tools. So linking what we do with the companies is very, very important. We also had a meeting in McFruit in Rimini, that is the biggest Italian fair, where we met with a list of colleagues of the project. And later we went in a packing house, one of the biggest packing houses. Most of the people that are here were there. So, and it was a great opportunity of interaction, because we need to take care to the practical part. We don't have to stay into the rooms, onto the desk, just on the papers. We have to write the papers, of course, because this is knowledge that we share with all the others. But it is important to interact with the people that works in the field, see which are the problems, and try to face them. For example, some colleagues took the box that is on the right. And thanks to all the people, also Anna Maria took some samples, Gianni Ceredi took some samples, and so on. And we analyze all of them. See, you see, there are more than 10 diseases, maybe also 15. And each fruit has its specific disease. Some fruit has more than one disease. So it was a very good opportunity of interaction, and we talked about how to approach and how to reduce this amount of disease. The meeting of today is a key point of this project, and it is. I'm very proud of Bashir that organized a very, very important meeting, and I'm curious to see how it goes on the interaction. And I followed on Facebook the first part when we were in the airport in Rome. So now whatever we are doing is in the internet. So a lot of people will see this video of this day on Facebook and can click, can share, and so on. So we will work, we are promoting what we do on different social media. And another meeting will be in Italy, for example, in 11 July. So in less than 1 month, we will have another meeting where we will meet and we will discuss together with a practical part. So together with a foundation that works with those kind of fruit, the managed fruit, and we have to find another opportunity, because this is a way to face the problems and to see how we can mitigate a little bit. This year, it will be an international congress. It is a scientific congress, and we will take, as we did for the Post-Harvest Congress, we will have a session, a dedicated session to our project on the first day of this project, where all the speakers will take their novelty to all the scientific communities. So, we are on the social media, so we are on Twitter, we are on Instagram, we are on Facebook, we are on LinkedIn. So whoever is curious about the information about the project, there is a website. They like this website because it was hacked, so we changed the website actually. Then this is stopandwaste.net. This is the actual website, and we are going through the use of, with the same partners, of safer tools within another project called Fresco Basics, that is related to the basic substances. It's something that we eat. Marwa will tell something later. And we will meet soon after the Congress in Lyon. We will meet in Paris to have the discussion about those. Now we are collecting the data with all the friends that I thank for all the work that they are doing. And last born is a cost action, that is a project of interaction where people from different countries of the world, more than 40 countries, are involved, including the unit coordinated by Bashir. And the theme is the same: sustainable network for agri-food loss and waste prevention, management, quantification, and valorization. This is called Food One Stop. And we will meet with Luis and with all the other people. Some of them are looking at us in Brussels, the 21st of September, because we will launch this new option, and it will be a chance. And any of you that is interested, we are getting a tremendous interest with a lot of requests of participation, so everybody can participate, and you are welcome to join this cost action, and more or less that's it, and thanks you all for that attention.
Merci, Professor Gianfranco. Thank you very much, Professor. I will give you a little present souvenir.
Thank you so much. Thank you.
You can. We can make together with Luis. Please.
Come into the picture.
Please, Marwa, please. Okay. Thank you very much.
Thank you so much.
Thank
Very
you.
kind.
Thank
Thanks
you.
for what you did.
Thank you.
Yeah.
And there is no. Plug in again.
Yeah.
Now we have.
Okay, Romana, say moment, a second. Okay.
Présentation faite. T'as une présentation. Merci. Nous passons maintenant à la présentation du docteur Marwa Moumni, qui travaille avec le professeur Gianfranco Romanazzi à Ancona, en Italie. Et ils travaillent ensemble donc sur les problèmes de post-récolte. Elle va nous présenter les travaux de recherche concernant ce qu'ils ont à Ancona dans le cadre de ce projet, s'il vous plaît.
Hello, everybody. I'm so excited today to be in INRAT. So it was a long trip, but it's okay. We reached it. So we are here to show for you how we used chitosan and essential oil and also ozone to reduce the waste that explained Prof. Gianfranco Romanazzi. So brown rot, it's a disease caused by monilinia, and it's the main disease of stone fruit. And this disease can appear in the field stage, but also in post-harvest stage in the packing house, and can cause waste can reach 80%. So these important pathogens, it is caused by three main species that which are Monilinia fructicola, Monilinia laxa, and Monilinia fructigena. So under the Stop Med Waste project, we use ozone to control this brown rot on the pitch, and we use the concentration of ozone 50 ppb during the day and 200 ppb during the night for 20 days, and we stored our fruit at 4 degrees Celsius. And to compare the effectiveness of ozone, we have fruits stored without treatment. So the result showed after 20 days, as you see here in this graph, at 4 degrees Celsius and exposed to 7 days shelf life, that the fruits stored with ozone is less contaminated with monilinia. And here you can see that we have used two sprayers, sprayer 1 and sprayer 2 in the pre-harvest treatment, which is the difference. Sprayer 1 is normal sprayer that it is conventional, and the number 2, it is innovative, that is able to switch on and switch off the nozzle automatic to reduce the quantity of pesticide in the field. And we have found that statistically, there is no difference between the fruit harvest from the sprayer 1 and sprayer 2 for the incidence of brown rot. As we know that ozone is able to reduce the microbial contamination, but to prove this, we have done a study to detect the gene and to see the gene responsible for this extant shelf of fruit. Our result showed that the ozone is able to inactivate the enzyme linked to the respiration of the fruits. For this reason, ozone is able to slow down the respiration of the fruit and extend the shelf life of the fruit and vegetables. So you have seen this slide with Prof. Gianfranco Romanazzi, but to explain more, ozone or also other alternatives and sustainable product, now all the researchers are focused on to be under the umbrella in Europe, under the umbrella of Farm to Fork, or also to reduce the use of pesticide, as he said, Prof. Gianfranco Romanazzi, because the supermarket, they are asking to reduce the amount of residue on the product of fruit and vegetables under the threshold of maximum residue level. So here we have basic substance. What is basic substance? Basic substance is a foodstuff. Foodstuff, it means that also we can find it in our kitchen. As you can see here, it's not so clear, but you can also see our review online. So, for example, cow milk, urtica, vinegar, and whey, it is used on food, but also used for plant protection. And this basic substance is approved in Europe, and we have 24 basic substances approved. And these basic substances are without neurotoxic effect. But unfortunately, it's still poorly known by phytosanitary consultant or also by researcher or agronomy. So this basic substance, as we said, is a foodstuff and used for plant protection. The first basic substance approved in 2014, it is chitosan. Chitosan is a biopolymer derived from chitin, and this chitosan, we can find in the outer part of the fungi. So when we treat our plant or our fruit with chitosan, the plant thinks that there is an infection of pathogens. In this case, we improve the defense of the plant before the real infection. So chitosan has triple properties. It is, as we said, is able to improve the defense of the plant or of the fruit, so has the properties of eliciting. It also is an antimicrobial, and also it produces a film when we treat with the chitosan or the plant or the fruit. Chitosan, it produces the film to cover. Here, as you see in this review, chitosan also, it is used as edible coating because it is safe. Chitosan we can also find on the pharmacy. It is used for diet. So chitosan, many research are focused on to use as edible coating, as you see in this orange graph. So in this recent year, we have okay. So in this yellow here, chitosan used as edible coating, and this orange, it is for all the research of edible coating. So there is an interest of work in edible coating. Also, Prof. Luis, he explained in the morning many edible coating. And in this table, you can see which are the chitosan available in the supermarket. So all of this list, it is available on the supermarket now, the commercial name of the product of chitosan. And Edible coating, the main properties of edible coating, it is the reduce of the respiration of the fruits, also to reduce the amount of ethylene. So all these properties, the goal is to slow down the ripening of the fruit or vegetables. Also, as we said, to induce the host resistance and to extend the shelf life. All these goals mean to reduce the waste of fruit and vegetables. We have this study that we done using chitosan and essential oil to control anthracnose on papaya fruit. In figure A is the control. Figure B is like using just chitosan at concentration of 0.5%. Figure C, it is the use of the essential oil Rata gravilans. And the figure D, we combined chitosan and essential oil. So as you see here, the control with the combination, it was the best treatment to reduce the amount of anthracnose on papaya fruits, but also to increase and to improve the defense of the fruit and to improve the enzyme linked to the mechanism of defense of the fruit. Another main pathogens for strawberry is gray mold caused by Botrytis cinerea. These pathogens can also appear in the field and stay in latent phase until post-harvest. And this is the typical symptom of Botrytis cinerea on strawberry fruits. So this disease, as we said, can appear from the pre-harvest and stay in latent phase on post-harvest until he found the good condition to grow on the fruit of strawberry. For this reason, we have this study that we treat our plant of strawberries before harvesting with chitosan and with phosphoric acid plus micronutrient and also with sweet orange essential oil. After treatment, we harvest our fruits and we store it 7 days at 4 degrees Celsius, followed by 5 days of shelf life at 20 degrees Celsius. The result showed that chitosan is able to reduce the amount of Botrytis cinerea on the strawberry fruit by 35%. So you can see here the effect of chitosan and compare to the control. Another study we have done using also chitosan and another product based on chitosan, it is Kosoga, to control the main 3 species as you have seen in the first slide of Monilinia and also Botrytis cinerea. The in vitro study was done using 6 concentration from 1 to 0.025%, and we mix our product with the PGA. And then we incubate our Petri dishes at 20 degrees Celsius, and we measure the mycelia growth. The result showed that the 2 compounds are able to produce inhibition of 100% of the mycelia growth of the 3 species of Monilinia in different concentration. So we have a total inhibition in Monilinia, and for Botrytis cinerea we have a total inhibition using Kosoga, and we have some growth, but also we have registered some inhibition using chitosan for Botrytis cinerea. As you see in this map heat, so here, using chitosan, the 2 concentration, the yellow color, it's 100% of inhibition. So Botrytis is inhibited 100% with Kosoga at 1% and 0.5%. And you can see that botrytis was less sensitive for the two products in the different concentration compared to the 3 species of monilinia that were more sensitive to this treatment. Another study, we have studied the volatile organic compound of 10 essential oil to control the same pathogens, botrytis cinerea, and the main 3 species of monilinia. Here you have the list of the 10 essential oil. The last one, it was a mix for 4 essential oils. And we have used a prototype designed by a postdoc in our lab. It is a specific prototype to study the effect, the volatile effect organic of compounds of the essential oil. So in the base of the plate, we have put the slide microscope here, and we put the pure essential oil. Then in the center, we have, this is the prototype that he designed, Samuel, to link the 2 parts of 2 Petri dishes. So in the base, we have the slides with essential oil, and in the top, we have the PDA with the plug of the fungi. The result showed that the minimum inhibitory concentration, MIC, it was less, lower, or using oreganum. So the effectiveness of oreganum, it was the good one. And thymus. So, but we can see also that botrytis cinerea was less sensitive for this essential oil compared to the 3 species of monilinia. So, also to conclude, in this recent year, there is a very good interest for study under and use of resistance for the fruit and plant. So, you have seen this also figure for the apple that asked how long will the induce of resistance protect me from the pathogens, because the fruit is under battle of list of stress and pathogens and search for an equilibrium between the resistance and the infection. So using as chitosan, ozone, essential oil, all these alternative products able to improve the defense of the plant or the fruit and reach this equilibrium between resistance and infection. All the stage for treatment is good, but earlier we treat, we can have more better result and we can have more extent of shelf life of the fruit and vegetables. Thank you for your attention.
Thank you very much, Marwa, for the presentation. Please, Marwa, stand. Maintenant, nous allons à la présentation online. J'espère que tout va bien online. With our colleague from DECO in Spain.
Hello, everybody. I'm going to share my screen.
Hello, Julio. Thank you very much for coming here and with us.
My pleasure. My pleasure.
So you can start your presentation, please.
Okay, okay, just a second, please.
Okay.
Okay. Okay, first of all, thank you very much to Professor Bashir and INRAT for the organization and for inviting us to share this presentation with all of you. This is Julio Marin from Deco Ibérica. I'm the commercial manager for North Africa. I'm located in Valencia. I'm agronomical engineer from the Polytechnic University of Valencia, and I'm going to talk about Deco innovations for the post-harvest industry. My presentation has a more corporative profile or commercial profile, and I hope that you enjoy. First of all, let me introduce Deco. Deco is a UPL company focused on the development and implementation of post-harvest technologies for reducing or avoiding the food waste. UPL is an Indian multinational company focused in the manufacturing and marketing agrochemicals. It's the fifth largest agrochemical company in the world. And to UPL belongs several companies, and OpenAg is a networking to link all UPL companies. We have NPP, which is a natural and plant protection, which provides biosolutions for crop protection and nutrition and soil health. We have NuturFarm, which is a digital platform to connect farmers. Vetopharma, which is a veterinary pharmaceutical laboratory dedicated to offer innovative solutions in the world of the honey bee. Okay, Advanta is a leading seed company, and Deco, as I told you before, we are a post-harvest company. We are more than 90 years in the market, and our main aim is to reduce or to avoid the food waste. Deco is a global post-harvest leader, and here there are some figures that I would like to share with you. I think are interesting. For example, each 10 citrus harvested in the world are protected by Deco. The 50% of the pineapples exported also are protected by Deco, and the 70% of the cherries. We are not only in those crops, we are also providing solutions for potatoes, table grapes, many types of crops, always with the same aim, to bring more and fresh fruit for the world. We have a global presence, which means a global knowledge about our field. Here in this world map, we can see that Deco, we have companies all around the world. We have a factory in Valencia, in Ibérica, where I'm in this moment. Also, we have a factory in Italy, another one in Israel, South Africa, Brazil, US, another one in China. And in Tunisia, for example, we are working with a company called Postagri. Probably you know all of them, and Mr. Jamel Benabda. And also we have a division in Egypt. I have seen that some of the attendant people are coming from Egypt, so we have also a division in Egypt. And here, I have, you can see just some of our clients. For example, in Spain, we're working with Frutinter. All these clients are big packing houses in general, in Spain, for example, Frutinter, Tecasia in Turkey, Mehandri in Israel, Hudup in South Africa, Titromax in Argentina, or Sun Pacific in the US. As I commented before, with OpenAg, with UPL Deco, we are able to cover all the steps of the food chain. We custom-made solutions for each of the steps of this value of the food value chain. So we have solutions from the farm to the fork. We are involved in the pre-harvest, in the packing houses, solutions for storage, for transits, for the distribution. So we are able to provide solutions for the full food value chain. Okay, and one of the first steps in this food value chain is the field. The fruit quality starts there in the field, so a proper agronomic method and approach is required. I have highlighted this solution. It's called DecoScreen. This is a calcium carbonate. It's a liquid solution that is, for this reason, it differentiates a lot to respect the competition. And this is a sun protector to reduce the sunburns and in the fruit and improve its quality. We have organic farming registration. With this solution, you can control the evapotranspiration, and also it's a physical barrier against insects, for example, the mayfly. Another of the, once we go from the field to the packing house, the first thing is the cleaning and disinfection. Cleaning and disinfection is a mandatory process. Unfortunately, maybe because of our daily work and we are too busy in the daily basis, we are not paying enough attention for the cleaning and disinfection. And it's one of the most important actions to do in order to get a good result and also to reduce resistance. Here in this slide, we can see one of our sanitizers. It's called Decobio. This is a hypochlorous acid. It has no residue at all. And we apply this product by atomization. And for example, in this video, I'm going to switch on the video. This is the damper area of the packing house, which is the most dirty place in the packing house. So we apply this product through this column with this atomizer. And in this graph, we can see how during the past time, the efficacy of the sanitizing and of the environment is increasing. Another sanitizer that we are highlighting now is our decoaerosol technology. The decoaerosol technology, this is a total discharge sprays, are very comfortable to use. The efficacy is very high. And this one is the active ingredient is potassium sorbate, E202. So with this product, with this aerosol, we can use it with fruit in the cold room. So from one side, we are making a disinfection of the environment, like we can see in this graph. Okay, that there is a big reduction in the contamination, microbial contamination in the environment. But also we are doing a treatment to the fruit. This other aerosol is the aerosol ultra. This one is based on glycolic acid with no residue at all. Also, it's a high spectrum biocide. This product must be used without fruit inside the cold rooms. And the efficacy is really very high, as we can see in this graph. Okay, the environmental microbial charge reduction is drastic. From 0 to 4 hours, we see very good results. And there is one very important advantage, which is the safety period. The safety period is just 3 hours. So after 3 hours, we can open the doors of the cold room and go into without any problem. Another sanitizer to highlight is our DecoSan. Our DecoSan is a HPPA. This is a peracetic acid plus hydrogen peroxide. This is a non-residual product also. In fact, we have this product with registration for organic farming. And there are many places to be added. Okay, drenchers, flooders, sprays online. And with this tool, with this solution, we are able to control the contamination and to control fungus also. In these graphs, this is a stone fruit, okay? And we can applying the product, we can see the comparison. In light green is the control, in dark green is applying the decosan against the monilinia and against the rhizopus in plums, nectarines, and peach. So you can observe in this graph that applying the product, we can reduce, avoid, even almost control these two pathogens. Another place to use it is as in the water, to sanitize the water, for example, in the drencher or in the treatment tank. Okay, here we have two plates to see the efficacy of versus Penicillium digitatum and bacteria. In the just in water, not applying the decosan, we can see the bacteria and the Penicillium digitatum growing on the plate, and applying the decosan, okay, the plate is empty. Another step and important for the post-harvest process is the coatings. The coating is the physical barrier against the fruits aging, and we develop solutions, tailor-made solutions, especially adapted to your packing house and your fruit condition and destination. I have gathered three of the, I think, most relevant in this moment. One is this coating. It's called Tetrasign Vegan. It's based on carnauba. And okay, we have vegan and organic certification. With this product, it controls really good the weight loss, delays the aging of the fruit, and keeps the fruit freshness for longer. And now this thing about vegans is a trending topic, and we are giving a lot of solutions for this kind of markets. The Citrasign NP is a coating formulated with pyramethanil. It's the first coating with registration and mixed with pyramethanil. The pyramethanil is a low-risk toxicologically fungicide. The MRL is high. We have in Europe 8 ppms. It's a very good tool for long storage of citrus, and also it's an excellent tool to avoid to fight against the resistance to the methyl for the spores. In this graph, this is the efficacy trials. And okay, comparing the results versus the control, the efficacy is very high. Another solution is the CoProtect. These are fatty acids. Okay, it's a fatty acids based product to be added in the water and in the wax. Okay, in the coating. In this, why we use this product? We use this product mainly to reduce or avoid the tearing injury. When we are obliged, or when we are requested to do cold treatment, we need to protect the fruit, okay? Because we are putting our citrus at 0 degrees. So here in this experience, you can see with Orri variety, we have the control. We have Golf 152 is a polyethylene plus shellac coating wax, and this coating added with the CoProtect and TVSet. As we know, TVSet, we use the TVSet as a skin protector, mainly in order to reduce the tearing injury. So with the CoProtect, we can get the same results as with TVSet, but with one advantage, that this is we are not adding an active ingredient. So for all those clients that they are requested to reduce the number of active ingredients, this is a very good tool, and we are using since several years for our clients. Another important thing, of course, companies like Deco, we are always looking for new biosolutions and new sustainable solutions. Fungicides now is the most effective decay control solution. The food safety and decay control solutions are our best ally to minimize the food waste. And here I'm going to sum up some of them. Decofito is a potassium phosphonate based product. It's a soluble liquid, and it has a very good synergy with the rest of fungicidal solutions. Okay. Also, the residue is part of the phosphatidylal. So it's effective against Phytophthora. And also, we are not adding a new ingredient in the packing house because this residue also is used in the fields. Another biosolution, this is a new biosolution by Deco, is Nexi. Nexi is a biofungicide for post-harvest treatment based on Candida oleophila, which is a yeast, okay, that this is isolated from the golden delicious speed. How it works? It works competing with the pathogens for the food. Okay, there is no MRL, and okay, what we can do is to increase the. This is a tool mainly for the organic produce. Okay, for the ecological market. And we can increase the marketable organic fruit, the availability, okay? Without, for example, this is Gala variety and this is Crispy Pink variety. So we can increase a lot the fruit marketable using the Nexi or not. And also there is a lot of synergy with conventional post-harvest solutions. Another very interesting tool is our smoke generation, our smoke generation technology. It's called Ecopir. This is a smoke generator based on pyrimethamyl. We have this product registered for many types, for many crops, persimmon, citrus, palm fruit, stone fruit, many of them. And okay, we can use it as a reinforcement treatment for long storage when we have in this case, for example, we have our pears stored and we need to store longer and we need a reinforcement treatment. So here we can see the efficacy in botrytis, against the botrytis using the Ecopir. And in this case, this is also a complement for an aqueous treatment, okay? So also in this graph, this is the variety golden delicious against botrytis, and we have a lot of efficacy versus the control. And another step and the key factor is how to apply the products, okay? Without constant dosage and proper post-harvest practice, any treatment is effective. With our equipments, the treatment is delivered homogeneously and uniformly and with precision and efficacy. And now I'm going to make a glimpse of our application technologies. The co-washer is an equipment that we install in the washing machine. And with this system, we apply accurately and we dosify perfectly the detergent and the disinfectant. The CODOS is our automatic wax or fungicide dosifier. With this equipment, we dosify the wax or the fungicide, and also we can, in order to regulate the dose, we can work counting boxes and pallets that are dumping on the line, or by artificial vision. The CODAF is our automatic dosing system for the drainer or the treatment tanks. With this system, we apply uniformly the fungicides and the disinfectants to the water. By this way, we keep the concentration of fungicides constant during the whole time. The co-central is, okay, all our equipments can be connected through the internet, okay? And we connect all these equipments with this system, the co-central, in our computer, in our tablet, in our smartphone, to check all the parameters and also to modify whatever we need. And the DECO SRC, this is a filtering system patented by DECO long ago, in 2010, something like that. And okay, with this system, we are able to don't drop the fungicidal solution of the drencher or the treatment tank. With this system, we are able to keep to don't drop and to don't waste water and to reuse the water until we want. And this last system is our DECO control system. Okay, with this system, what we do is to provide, in a cold room, we put all the devices in order to control all the parameters for storage, for cold storage, temperature, CO2, humidity, but also we add devices in order to transform the cold room into a degreening room. So we are able to control and to monitor the degreening of the citrus. And lastly, I would like to say that, okay, all the products are very important, also the equipment to apply the products, but at the end, it's very important to provide a good service in order to be able to monitor and to make the follow-ups and to take all the data to take decisions. And this is what, okay, our multidisciplinary team is doing every day. So we provide a lot of services to our clients in the quality control, for example, fruit quality check or regular visits to check all the process in the packing house, laboratory services also. For example, we monitor residues in fruit, a microbial contamination in the line or in the environment, etc. Also, we do monitoring of resistance strains. And also we make tailor-made advice to all our clients. So thank you very much. And if you have any query or any question, please let me know. Thank you very much for your time.
Thank you very much. Julio, thank you very much for your presentation. And I am happy for the good communication online with the Zoom communication.
Yes, perfect. Fortunately, now it's perfect. At the beginning was a little bit complicated, but now it's excellent.
Thank you very much for your presentation.
Thanks to all of you. Okay.
You will you follow with us the rest of the presentation, please.
Yes, for sure. Okay, thank you.
Now, now we have some presentation from companies. Maybe Si Hassan Ghidawi. Monsieur Tariq Tirahir, if there is another one that he will participate from Bioprotection, you will present something oral or with in the you see, we have Hisham Onalla, the first one. You will vous voulez présenter maintenant? Ah, alors, parce que je vais suivre ce qui se trouve ici. On commence par Monsieur Hassan Ghidawi. Please, Monsieur Hassan Ghidawi. Monsieur Hassan est le gérant de la société Fertiplan Pesticide Company. it is a testimonial from the professionnels. Il va nous donner quelques... oui. خليها ساعة جيت اوكي اوكي البر يسمعوا فينا زيادة البر يسمعوا فينا اونلاين زيادة Monsieur Hassan Redaoui, merci d'être présent avec nous. Tu nous honores par votre présence et donc je vous passe la parole pour donner de votre expérience et d'autres savoir-faire sur le terrain maintenant, c'est pas au niveau des laboratoires et je vous passe la parole.
Merci beaucoup. Merci beaucoup, Sibéchir. Merci pour l'invitation. Je me présente, je m'appelle Hassan Redaoui, je suis de formation ingénieur agronome, diplôme des études approfondies en physiologie végétale. Je suis
I work in a Tunisian company, Fertiplas, specialized in pesticides and fertilizers. I have been working in the same field for 30 years. So I take it or seize this opportunity to present a product, one amongst the products, which is in the conservation or the shelf life, to extend the shelf life of the produce. It's a product from a Czech company. So the active matter is the Pythium oligodendron. I very quickly will introduce the product, the Pythium oligodendron. So the active matter is the Pythium oligodendron. So it's a sexed and asexed reproduction. The optimum is 30 degrees and 77, maximum 37 degrees. It's close to the variations of the different fungs which are in the field. We are talking about a Sush M1 that is wild, extracted from the soil from the Czech Republic. It is not modified genetically. It's a fungi that is comparable to microorganisms present in the soil with weak concentration. It parasites on the microorganisms, fungi, and decreases resistance. It is used as an active matter for the protection of plants and vegetal entities or plants. Morphological characteristics. Sorry, I need to find the appropriate slide. So the Pythium oligodendron is the class of Ironzod 7, and from the family of Pediaceae, the gender is Pythium. The genre is Pythium, or the Pythium oligodendron. It's a fungical, biological fungus with a large spectrum of cryptogamic diseases. Sorry, there is a technical hitch. It has got 3 modes of action: microparasitism, self-defense of Eurygandrium, and the synthesis of tryptophan. It's a mode of stimulation of growth. Very quickly, the mode of action, microparasitism, we are all familiar with it. We are talking about the Oligandrum endogrim. So there is an enzymatic degradation. So it decreases, the activities of the fungus, and it kills it. So that's one example. The Tetanium religulum acts on pathologies, it acts at the level of the degradation of enzymes. Of course, we have different. Here, this is at the beginning of the test. At 1 minute of 14 seconds, we have the attack of this Pythium oligandrum. Then it will end up after 5 minutes. It ends up killing the parasites. Another example of mode parasitism action is the action on the Phalophora, which is a family of the Monolia fungus. So here, for example, we have on a tomato, on a potato, sorry. Once we apply it, we will end up having a total destruction of the fungus from the family of Monolia. Another example of action mode is on the So the same thing here, the Pythian oligandrum will end up, as you can see here, by completely inhibiting and destroying. So in summary, so we have the mycoparasitism, which is of course adapted in a certain category of fungus, especially the Cryptogrammic one, the Ophthalmaria, the Botrytis, the Fusarium, the Verticillium, and the Scrutonia, and the Gadium anomyces. So it acts on these fungus. These are amongst the fungus where the Pythian oligandrum is more efficient. The second mode of action is through the synthesis of the oligandrin, which is the protein that enters within the self-defense system of the plant. So an example, the oligandrin, because it does not, it has some specific characteristic. It's the protein that is the most acid, like the lizard one, which enters through an infiltration. So we have the oligandrin, we have a protection vis-a-vis the aggressive samples of fungus, the cryptogamic, like the Phytophthora, Fusarium, the Botrytis. So it is the first protein, elicitin, like that is totally devoid of phytotoxicity, and while having an activity of resistance that that stimulates resistance. This is another example, needless actually to look at it. For the mode of action also of the antibiosis is the most adapted against the following fungus: the Oxidomonas, the Dicina aspe, the Restonia plurissum, the Titulia cari, and Leptosphaeria maculans. The third mode of action is of the growth stimulation. So the Pythium oligandrum, it secretes at the level of the radicals a molecule, a vital molecule for the biosynthesis of the auxin. So the action mode of this epithelium, the microparasitisms, self-defense, and the stimulation of growth. This is the commercial product, as you can see here. We have had an official recognition or homologation in Tunisia. In Tunisia, it has been homologated on Syria against the Fusarium, the Septoriose. On the botrytis, there is an homologation in progress. It was homologated on the strawberries, on the tomatoes, like, for example, the anthracnose and botrytis. It is homologated for the dates, which are fungus that are responsible for the drying of the palms. But for the pomegranate, we are still waiting for an official homologation. We have already reached very positive results on the grounds against the Fusarium, the Didactylium, the Septoriose. But normally, let's hope that we will have very soon the homologation. Then we will, of course, conduct further studies on the shell life, and why not having some official recognition and homologation on other fruits and vegetables. We have some scientific assays or the use of polyvarsin. It can be used through the coatings of by either the coating or by immersion. It can be also in the warehouse, and it can be applied also after the harvest to extend the shell life. Here, I would like to share with you some results that are related to some diseases during the shell life. Here, this is the accreditation that we have received for the application of shell life. We use the principle of brumization. Why? Because it is a new biological technology which is modern and without residues. This is a treatment that is profitable. The method of nebulization, allows a better covering of the surfaces of the fruits and the vegetables which are treated. The other advantage, if we can use a feeble quantity of water that does not increase the humidity and that functions in very good storage places, which is this is actually the best way, what we call the fogging way or the brumization way. So, as Mr. Depot has already talked about the tools or the technologies that we use to treat. There are some, of course, we need to have foggers that are of a biotype that should be used. As you can see, we will see later on in a video. The glycerol added to the application solution should not exceed 5%. Application uses of a size of 12 to 15 are recommended for a nebulization by polyvarsin, which is new, and a constant agitation with the recipient of biopreparation is necessary. Here, an example of the technology that was used, an example of this technology. Here you have the technical details related to that device. The pulse fog bio is used now. K22 bio is used, and it's the most efficient one, as you can see here. The application spectrum, then the parameters in this technology, or this technology actually is convenient to a variety of environments. The storage technologies are very simple and they are really cutting edge technologies. the technique that was tried in the conditions of the field. For example, for the protection of the strawberries and other fruits approximately in the harvested sites. And it can be used easily combined with the storage technology, the most modern storage technologies, which include the combination of the freezing and refrigerating of the ULO, ultra-low oxygen, and the inhibitors of maturation, like the Smart Fresh. Protection of cultures, agriculture against the infections of the fungus infections in general and specific to their origins. We have here some of the products that over which we conducted our tests. Here are the practical results on the apples, as you can see here. The control. Here is the polyversant, and here it's an association of apple botrytis and apple bitter rot. So the mixture or the association here, the effect of polyversant, sorry, and here the association. Here, this is against the botrytis. Apparently, here it's an association of polyversin and the bellis. Here we have some practical results conducted on the field on the strawberry field. Of course, we have conducted some molecular analysis on the penicillium, the botrytis, and the Totrichum on strawberries during storage at 15 degrees Celsius. The K, you've got the control, ETC. So we had one day, 3 days, after 7, 9, 11 days. So you have all here the results collected. Here are the results, the practical results of molecular analysis of penicillium on the strawberries during the storage. Here you have the samples 1 day, after 5, 7 days, 9 days, and 11 days after. So you have here the examples. They are not part of the test, but it's a scheme pattern on the strawberries. You see that the result of the polycerium after 11 days, it remains efficient against the penicillium on, of course, the strawberries. After even 11 days, it is still efficient. Here you have the botrytis. Here you have 0 day, which means the harvest day. It goes up to the 11 days. There is a neat difference between the ones treated with the botrytis and the sample collected. Here you have some other assays with the collectrum, the collectorictum. You see here from 0 day, which is the harvest day, and 11 day, and you see here a neat difference between the control and the polyversum. The practical results will also on the field of apples. We have had 400 kilos on box, 200 grams of polyversum with 4 liters of water. And so you have here the after and before. These are examples of pictures of standard treatment and treatment with polyversum. So the polyversum with control, as you can see here. The effect of polyversum on the storage, on long-term storage of apples. Here, the assay or the test, sorry. The first test was conducted in a frigorific or frozen room during 6 months. Then a storage for 4 degrees during a period of 6 months. And then in a refrigerator, 28 days at 6 degrees Celsius and a humidity of 70%. Fogging treatment with 150 grams of polyversum, distillate water for 0.5 plus 5% glycerol and 0.1% of calcium chloride dehydrate. Here, the room with control of the oxygen, the storage room with control of oxygen. Here, the loss of the harvest in the apples, according, of course, to the diverse methods of storage. After 175 days of storage in a cold room in condition ULO plus 28 days of storage in a refrigerator, as you can see, the cooling chamber control with the polyversum, as you can see, it has, as you can see here, you have a neat difference between the cooling chamber control and the polyversum, even with the cooling ULO control. You can see the neat difference between the results. Here it's a summary. The damages of the fruit have been significantly reduced by 41% in storage and cooling rooms. And so there is a difference of 59%. We have reduced by 41% of the waste in a classical cooling chamber and 51% in a ULO. So as you can see here, the results, the conclusion, the protection, post-harvest, protection of the fruits, vegetables, and other cultures through the fogging represents a very interesting option in the variety of other applications. The polyversum has proven itself to have a beneficial control against the propagation and the dissemination of fungal sicknesses or maladies, diseases, the effects on other characteristics on fruit like the color, etc., has also been proven themselves to be efficient. So thank you very much for your presentation. We will hand the trophy to you. Thank you very much.
Okay, two quick communications. They're not going to take much time, just 10 minutes as mentioned. So Ms. Hanin Ayedi, representing the company Bioprotection, the company responsible for the sales of pesticides, and they have certain things to say concerning the decay of fruit and vegetables. So the floor is yours. Thank you very much. Before tackling the tablet, so Hanan Ayedi, rural engineer, and for the time being, I hold the position of the company Bioprotection. So those who don't know, Bioprotection is a company enterprise specialized in the commercialization of distribution of all fertilizers, and we collaborate with many international companies, the international companies Syngenta, BASF, the German ones, the multinational Indian ones, and many other companies concerning fertilizers and fertilization. So rapidly, my presentation of my company. I'd like to thank, first of all, Mr. Bashir for the launching of this topic, which is quite important from our expertise with the farmers, we noticed that the post-harvest diseases now more and more highlighted, and as it was done by the researchers as well as the colleagues concerning the rate of waste reaching 30%. On the one hand, you have the constraints and the demands of the international. Now there are demands, there are requirements that do exact, and they are more and more rigid. Hence, the idea for a company in collaboration with our supplier, Sanginton, to put on the Tunisian market a solution specialized in these diseases. So the product Scholar is certified on the Tunisian market, started in 2022 rapidly. Scholar, without giving too many details, Scholar, it is the expert solution for an advanced protection. Scholar is a new fungicide post-harvest. You have a large spectrum fighting all these diseases, improving the quality and the fruitability of the work. You have 100 liters per proximil concentrated, and the action, the product is part of the product according to the classification FRAG, and it is the only fungicide in group 12, and it has an activities that is present. It is efficient concerning many diseases. Now, concerning the fluoxetine, it is classified by the American agency as a fungicide that is produced and with reduced risk and with a weak impact on the environment, with no danger on human beings, and integrated the struggle about this. Concerning the Scholar, it is a tool, larger spectrum. It is an action on the citrus, be it with kernels or not, potatoes, pomegranate, other fruits, and the Scholar provides the largest spectrum in the struggle against diseases. The mode, the action mode, so it is a protective action before germination of the plant. It is going to inhibit the risks and reduce it. As you can see, its application on the pathogen agents, allowing to mycelium to invade the different cells rapidly. The cells, as you can see on the citrus, it has a good action against Penicillium acnort, and a bit on the acid decay. It has a good action on the Penicillium. Concerning the fruits with kernels and with the three species. So if we might say it has a good flexibility for usage, and it is applied according to many methods, a treatment per dipping or with acid. So thank you for your attention.
Merci, Madame, pour votre présence.
Thank you very much for the presentation, Madam. So we will, like everyone, we will hand you the trophy. We'll give you a present.
Okay, merci, merci. Maintenant,
Thank you very much. Now we have Mr. Achira. He represents the G-Fruit company. He would talk to us about the observed issues in many enterprises or companies. And then later, if you allow it, we will have a Q&A session. If any among you has a question, so we will offer the floor for a Q&A session, and then we will close the session.
Bonjour tout le monde.
Good morning, everyone. I introduce myself, Tariq Era, Vice Director of the exporters from the interprofessional group. It is under the umbrella of the Ministry of Culture. It's managed by a steering committee. Sorry, a quarter from the UTICAF, 1/4 of the farmers, and 1/4 are representatives of the different ministries which intervene in the sector. Our mission consists in principally the promotion of quality at the level of the production through national companies of treatment, through the specific quality, through the management of certification of plants, the promotion of exportation through campaigns of exportation, for example, everything that is related to the preparation, the follow-up, and the evaluation, and the promotion of our products on the local and international markets. Among our other missions is the dissemination of information for, of course, the different stakeholders. Now, as far as this seminar is concerned, let's say, in how to extend the shelf life of a product, I will talk principally of the marketability. So, because the problem is how to ensure that marketability and how we make sure that there is a logical, affordable price for the consumer. So, our target is how to make sure we have our products with a very long or a longer, let's say, shelf life in good quality, and so on and so forth. And of course, with an affordable price. So we offer concrete solutions to extend or prolongate the shelf life of the products. We have a problem. One of our major preoccupations is the logistics. We need a minimum, for example, for the local market, sorry. No, the international market for the Gulf countries, for example, via maritime routes or other destinations. Actually, it can reach 7 days. For African countries, it can even go beyond that. We cannot export, for example, to the African market. We don't have any maritime connection with them, and so it takes a very long time. And we need to find solutions as far as these issues are concerned. Something else, so we intervene also to promote the use of pesticides. Here we organize, for example, a raising consciousness campaign to rationalize the use of pesticides so that we can respect the different norms. These are days which are destined to the farmers. There are some campaigns, we display the flyers to rationalize and encourage individuals to respect international standards and of course to raise consciousness to not only to the people who can to the farmers but also to the consumers, so that of course the consumer can have a good product with good quality and a healthy product. So we have, let's say, some techniques which need to be further developed and improved. We need to take into consideration the impact price, the price of these products. So the price, we need to pay attention to the pricing of these products. Thank you very much for your attention. Thank you, Mr. Taraka. Thank you for your presentation. And thank you for having informed us for what we need to think about to reduce the residues and take into consideration the distance of transport so that we can make sure that these products can reach remote destinations, of course, in a quality, with the high quality. So these are things that the researchers need to take into consideration by using different treatment products. So we need to take to bear that in mind while thinking about conducting research in this sector.
Okay, maintenant.
Now we open the floor for the Q&A session. If you have any questions, if you want further clarifications, we are at your disposal. We are all ears.
Ou bien si quelqu'un veut faire un certain commentaire.
If you have any observation or comment also, you're welcome. Before we close and go for lunch. Maybe Mr. Professor Manadzi wants to say something. Professor Manadzi, if you have something to say. Si vous avez quelque chose à rajouter. Je n'ai absolument rien à rajouter parce que déjà il est
little bit and to walk around. But a big thank to Professor Alagui because he organized an excellent meeting, and congratulations to all the presenters that I heard. Very, very good results, very good opportunity for the growers and for a better world to which all of us will have to contribute. Thanks to all the speakers that spoke at home. Thanks to Lucrezia D'Orchenzio that managed the online part through Zoom. And thanks you all for being here. Thank you.
Merci beaucoup. Thank you, Professor Romanazzi. Prof.
Professeur Louis, est-ce que vous voulez dire quelque chose? Si tout est clair, je pense que tout est clair. Everything that we have presented, I think, is clear. There are no questions, so I can safely conclude that we are all satisfied, so we can move on now to the lunch break. And thank you very much for your presence.
One question. Please. Excuse me. I'm curious to...
Je suis curieuse de savoir s'il y a des histoires à succès pour la conservation de la grenade ou des pommes avec des produits naturels. Est-ce qu'il y a des résultats probants ou... So if you want to talk about any success story, if any person would like to share a success story related to the conservation and the preservation of the fruits. Mrs. Sonia, believe me, quand j'ai commencé à travailler, je pensais que c'était une chose simple et que ça allait pas être sorcier. Mais après des années de travail, je me suis rendu compte que c'est pas du tout facile de trouver des solutions à des problèmes, surtout le pénicillium. On peut le trouver partout. Donc, l'isolation ou l'isolement de l'isolation, c'est pas facile, c'est pas sorcier, pardon. Mais traiter et contrôler ce pénicillium n'est pas du tout une simple affaire. Tous les produits qu'on utilise n'ont pas. Enfin, les produits n'ont pas tous montré une efficacité auquel on s'attendait au début. Donc, on s'est rendu compte qu'il y a énormément de difficultés, même lors du travail, on fait face à d'autres problèmes et on s'est rendu compte qu'il est très, très difficile de trouver des solutions et c'est la raison pour laquelle je me dis, bon, j'ai commencé depuis des années, j'ai commencé à travailler dans plusieurs pays et jusqu'au jour d'aujourd'hui, on n'a toujours pas trouvé une solution. Pourquoi? Parce qu'en fait, c'est sorcier, hein, c'est pas du tout simple à traiter, même en utilisant des méthodes biologiques de contrôle. Peut-être on devra travailler davantage sur les méthodes biologiques de contrôle. Peut-être que c'est les champignons on pourra travailler sur les champignons qui attaquent les insectes. Je vous remercie de votre présence. Je suis très content de vous revoir et de partager avec vous ces informations. Et je suis très content avec la réussite de la communication avec les différents pays. Donc, la connexion était importante pour nous en Tunisie. C'est important de voir ça en Tunisie et c'est positif. Je remercie encore une fois.